Workshop Chair, Director, MET Center for Nanomedicine. I request Professor Dr. Madhumita Ghosh, co-convener for this workshop. I request Professor Dr. Ashish Mani to please come to the dais. So I request Dr. Chansi, the convener of this workshop, to come to the dais. Thank you, everyone. So good morning, everyone. On behalf of our Honorable Founder President, Dr. Ashok K. Chauhan, sir, Honorable Chancellor, Dr. Atul Chauhan, sir, and respected Vice Chancellor, Dr. Balwinder Shukla, ma'am, it is my proud privilege to extend a warm and heartfelt welcome to all our distinguished dignitaries, eminent scientists, respected academicians, industry leaders, faculty members, research scholars, and the participants to this 3 days international workshop on from design to detection, targeted drug delivery, crossing biological barriers, and AI-driven biosensors, sponsored by ANRF. We are deeply honored by the august presence of Professor Dr. Trimurti Valpandian, who is the professor at Ocular Pharmacology and Pharmacy Division, Dr. R.P. Center, AIMS, New Delhi, and also Dr. Rajkumar Tulswani, scientist E, Biomedical Science, scientist DEPAS, DRTO, Ministry of Defense, Delhi, who will be joining us shortly. Honorable Founder President Dr. Ashok Chauhan, sir, Honorable Chancellor, sir, Honorable Vice Chancellor, ma'am. I'll also feel honored to welcome Professor B.C. Das, Chairman, and Hargovind Khurana, Chair Professor, MET Institute of Molecular Medicine and Stem Cell Research, who is the Dean of Faculty of Allied and Health Science, Professor Dr. Seema Bhatnagar, ma'am, who is the chair for this workshop and also the director for MET Center for Nanomedicine, MET Noida. So let's welcome all them with a big round of applause. Now I request Ms. Ashmita to take forward the proceedings.
Good morning, everyone. This workshop brings together eminent scientists, academicians, innovators, and young researchers to discuss cutting-edge advancements at the intersection of nanomedicine, targeted therapeutics, artificial intelligence, and next-generation biosensing technologies. It aims to foster scientific exchange, interdisciplinary collaboration, and innovative thinking towards addressing modern healthcare challenges. May I request all the esteemed dignitaries to join us in the lighting the ceremonial lamp, a timeless symbol of knowledge and enlightenment, and the triumph of wisdom over ignorance. With this, we would like to invite all the dignitaries to come forward and seek the blessings of Goddess Saraswati with Saraswati Vandana as well.
शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। तू स्वर की देवी है, संगीत तुझसे, हर शब्द तेरा है, हर गीत तुझसे। तू स्वर की देवी है, संगीत तुझसे, हर शब्द तेरा है, हर गीत तुझसे। हम हैं अकेले, हम हैं अधूरे, तेरी शरण में हमें प्यार दे माँ। हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। मुनियों समझी है मुनियों ने जानी, वेदों की भाषा पुराणों की वाणी। मुनियों ने समझी है मुनियों ने जानी, वेदों की भाषा पुराणों की वाणी। हम भी तो समझे, हम भी तो जाने, विद्या का हमको अधिकार दे माँ। हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। हे शारदे माँ, हे शारदे माँ, अज्ञानता से हमें तार दे माँ। हे शारदे माँ, हे शारदे।
Thank you, everyone. As a cherished tradition at Amity University, we extend a warm welcome to all our esteemed guests by presenting them with the saplings. So I would request Dr. Professor B.C. Das sir to welcome our keynote speaker, Professor Dr. Thirumurthy Velpandian, with the sapling. So may I please request you come forward. Now may I request Professor Dr. Seema Bhatnagar and Professor Dr. Madhumita Ghosh to welcome Professor Dr. V.C. Das with the sapling. Thank you. To share To share further insights into this 3-day international workshop from design to detection, targeted drug delivery across crossing biological barriers and AI-driven biosensors, may I kindly request Professor Dr. Seema Bhatnagar to deliver the welcome address and brief us about the objectives and vision of today's workshop. Professor Bhatnagar is a distinguished academician and researcher with extensive expertise in medicinal chemistry, small molecular drug discovery, and anti-cancer research. She has made significant contributions to the scientific community through her research. May I now invite Professor Dr. Seema Bhatnagar.
So with the blessings of Honorable Founder President Sir, respected Chancellor Sir, respected Vice Chancellor Ma'am, distinguished dignitaries all on and off the dais, esteemed speakers, faculty colleagues, PhD scholars, and dear participants. It gives me immense pleasure to extend a very warm welcome to all of you to this ANRF-sponsored workshop from design to detection, targeted drug delivery, crossing biological barriers, and AI-driven biosensors. Today, as we celebrate Technology Day at Amity University, it is only apt that this workshop brings together an inspiring community of scientists, researchers, and young scholars united by a shared vision to advance the frontiers of biomedical science through innovation and technology-driven solutions. We live in an era where precision therapeutics, intelligent biosensing systems, nanotechnology, and artificial intelligence are rapidly reshaping healthcare and redefining the future of disease diagnosis and treatment. And the theme of the workshop perfectly captures this transformative journey. I'm confident that the deliberations, discussions, and interactions during this workshop will inspire new ideas, perspectives, motivating young researchers to contribute innovatively to the field of healthcare and biomedical sciences. I express my sincere gratitude to our distinguished speakers and experts for graciously accepting our invitation to share their valuable knowledge and their experience with all of us. I also warmly welcome all the participants whose presence and enthusiasm adds great value to the scientific congregation. I hope this workshop will provide an enriching learning experience and inspire all of us to contribute meaningfully towards
scientific innovation, and societal well-being. With these words, I once again welcome all of you and wish you a very highly productive, engaging, and intellectually rewarding workshop. Thank you.
Thank you, ma'am, for giving us such a comprehensive overview and valuable insights into the sessions planned for today's workshop. Your address has beautifully highlighted the vision, objectives, and scientific significance of this gathering, creating great enthusiasm and anticipation among the participants. Now, it is our immense honor to invite Professor Dr. B.C. Das, Chairman, and Hargovind Khurana, Chairperson, Professor, Amity Institute of Molecular Medicine and Stem Cell Research, Dean Faculty of Health Allied Sciences, Chairman, University Research Council, Vice President, Amity Science and Technology and Innovation Foundation. A distinguished molecular oncologist and pioneering cancer research, Professor Dr. Das is internationally recognized for his remarkable contribution to cancer biology, tumor virology, and human papillomavirus.
I think it is still morning. Good morning to one and all. And first of all, it's a great occasion today for the Center for Nanomedicine, which is now become an independent center. Initially, I was associated with this creation of this center, and the Professor Tinku Basu was then the professor of nanotechnology at the Institute of Nanotechnology. She took the initiative to that I want to make a nanomedicine department or a center. But then founder president said it's a nanomedicine, means it's some relations with the molecular medicine. So honorable founder president said, okay, you come out as you are the faculty or the of AINT. So she came out and a new center was established and she became the director. But it was remained under the care of Amity Institute of Molecular Medicine and Stem Cell Research. Somehow that time I was abroad or somewhere. So I never knew when I came back. So I could come to know that this center is created and it is under. And it was very religiously till her death. It was being guided me and all aspects were looked after, but she was independent director. But whatever matter, it used to go through me and I used to fight for things and that. But now this center has come into existence under a new director. We had a hard time to find a director for this. So sometimes it was suggested that it can be merged with some department. So I was against of that the department cannot merge. It's a new discipline and there is no such the independent department or institutes in the country. They might be doing here and there. Nanotechnology is there, but nanomedicine maybe people are doing, but there is no full-fledged department or institutes in the country, in any university or anything. So I was, of course, it is always I am in favor of maintaining the discipline, like nanotechnology was about to merge or dismantle. I thought the discipline should continue. So that was always the thing, that discipline has its own importance. If it is something which is very not very important at the present context, then we can easily get rid of it. But which is very important, nanotechnology in everywhere, drug delivery, and there are huge number of applications. So that applications has been, the workshop is aligned to that. Before that, that it is all these things came into in the all types of advanced center and very innovative centers or the institutes have been established, including my own institutes, Amity Institute of Molecular Medicine and Stem Cell Research. It was founder visions, and with his vision only we could come to in existence, and that created a lot of new directions of research. And similarly, this department is doing excellent for the, I think she did almost 7, 8 years, or think about it, 19 onwards. So it was a good time. She is no more with us. So, and this is all because of our most respected Honorable Founder President, Dr. Ashok K. Chauhan, and also our dynamic Chancellor, Dr. Atul Chauhan, and Honorable Madam Vice Chancellor, Professor Dr. Balvinder Shukla. She is busy today. There are Technology Day, many meetings. And also I'm, before my was Professor Dr. Chandradeep Tandon was the additional Vice Chancellor who was to also speak. I do not know he will have time to come over here because there are a lot of other guests, and Chancellor himself has come in the I2 moot court. So this is, and also the most important is the today's guest speaker, the keynote speaker, Professor Tirumurti Melpandian. So I am meeting you first time, though I am a regular visitor to AIMS. Of course, after COVID, I am rarely visit, and because I whole life was in ICMR. So ICMR going means because all my contemporary faculty in AIMS are very closely known. Still, there are many closely known, and so ICMR, of course. So welcome to Amity University. I do not know you are first time coming or the first time. Earlier, it was great. So you are Amitians already. So who comes once, they become Amitian. So welcome once again. And all our colleagues on the dais, including the chair of this workshop, and all my colleagues in front rows and back rows, and all the PhD students and other students. So welcome to this the workshop inauguration. So this workshop is the from design to detection, targeted drug delivery, crossing biological barriers, and AI-driven biosensors. So there are biosensor was always there, but now it is the everywhere people are using the biosensor because it makes things easy and less time consuming and maybe perfect also. So without very hassles, it is being used now everywhere. And it's a very apt time that biosensor research and everything, many things have happened, but then it is an apt time to add biosensor with the AI device. Come this side here, front, Dr. Manoj, here, this side, here. So this is as you know, this is a workshop designed to develop a manpower, and there are most important is the participants. I'm told there are 25 participants from different Delhi NCR from also outside. So it's nice that those who are coming first time to Amity campus. So please find time, though it is 3 days, but you have weekend time, so you can go ahead with seeing the campus all around. And if there is any department of interest, you can visit the department. So welcome the most important person, those who are the participants. So this workshop, actually, it is the actual that nowadays the NRF and or DST earlier, this is to give the fund for the projects. So these projects has a one component that is called SSR, Scientific, Social, Scientific Responsibility. or scientific social responsibility means you can do the research in your four walls, but something you should contribute to the society. One can say whatever research we do for the humanity or benefit of the humanity or the society, but it is not that what we do the research, though it is translation research, it is not necessary that it will immediately come into use in the public or the society. It will take maybe year, maybe 10 years, maybe several decades. So it will come to the use of the public. So, but then immediate, people want something immediate effects or the impact on the society. So it is a nice thought of the ANRF, the agency which has come out from the DST, and they have made it mandatory that whenever you do a research, they give the additional fund allocated, not big amount, but sufficient enough to carry out some social responsibility. So, but science is there related to your research aspect. You disseminate that knowledge or share that knowledge or give that technology to the person you are getting trained, that you invite the participants, students or PhD students or early career scientists or the faculty, to give them exposure to build up the manpower in the area, specific area on which the project is built on. So this is a scientific social responsibility aspects. So that's why it is the workshop to train people. And this workshop was the Anusandan National Research Foundation or NRF project, and they give a grant. So there are, it was actually Dr. Chansi Gupta is the ACNF, ANM, the faculty who is having this project, and this is the part they are doing it, and also Madhumita Bose also involved with that. Of course, the director, Dr. Sima Bhatnagar is involved. So it's a small center, but it has a particular vision, and you can understand that nanomedicine means it could be drug discovery, drug delivery, and all other aspects, including biosensor detection, diagnosis. That's why aptly the title has been given, and that incorporates diagnosis and also drug delivery aspects. So this workshop is aimed to advancing the scientific training and critical domain in the drug delivery. And then with the help of the AI-enabled biosensor technology to bridge the gap between the laboratory innovations and real-world technological translation or their use. So this is a good the vision to do this workshop, and it's for a short 3 days workshop. You cannot do much because its fund is very less. But within that, I see that there are several good speakers have been invited, and some are from abroad also. So there are Professor Daipan Pan. I do not know when he is joining. He is joining today or tomorrow. So those who are in online, I also welcome them. Those who are on the, so I welcome some of the distinguished faculty like Professor Dr. Daipan Pan, who is from Dhoti, where Hook and Lloyd Hook, chair professor in the nanomedicine in the Pennsylvania State University, a pioneer in the clinically transferable nanomedicine platform. So that's great that she has been many times traveled abroad, and so she has a connection, so she could invite few of the people. Dr. Mariam Rahman, professor of neurosurgery, University of Florida, USA, and so she will be online. They will be online only. So time frame I do not know, but 3 days program, so one can attend. And I think that you should give the link also people. So those who can join, may not join, they can join and listen to them. And Abhinav Bhushan, Illinois Institute of Technology, USA, expert in the microfluidics and microfabrications and biosensor development. And then Kritika Rangarajan, is that right? Rangarajan, AIMS, Delhi, specialist in the AI data science onco radiology, and AI-driven medical imaging, and Himanshu Baveja with the Yashoda Medicity, speaking on the regulatory framework. So the workshop is also features the global industry leader like Sheetal Kaushik Bharadwaj, in vitro TEM from Netherlands, Vaibhav Garg, President Global Government Affairs, Merrill Group of Companies. So I'm giving some names I have taken, but it's a good collection or the accumulation of the experts in the different areas of this discipline. So, so it is a great pleasure in this that this institute I was associated or created in the beginning rather, and now she is handling independently. It's honored to be here and to share with my views that I once again welcome all the guests, but as the one of the associated with this department as well as the center, and as a chairman of the University Research Council, our primary endeavor is to at the Amity University to cultivate the research and innovations ecosystem where fundamental science translates into the societal use or a societal benefit. And that is what the SSR. So we are also, all these colleagues and from the biotechnology and the AIP, Amity Institute of Pharmacy, and my own institutes and microbiology, and they are all here, that we are doing research mainly to the benefit of the society or humanity. So this is one of the attempts to make it develop a manpower in this field and to make the people aware of the new development in the field. So this Amity University, in the spirit of this site of innovations, and I am incredibly happy that our leading scientists or researchers or colleagues have find it over, they have been doing excellent science, and that is why the Amity is known as the research and innovation driven university. It is truly so, and the technology day will be more, if you have some time, when you can go, then to listen that how it is. There are also several experts come in there, and one can imagine that we have almost 1,500 patents which have been filed, but more than around 500 are already transferred to the industry, and there are also many are transferred. So that also it is important to listen that there are others, those who do not know the Amity has the H index, that when you evaluate an individual scientist, we say, what is your H index? So that starts from the 2, 3, 2, 30, 40, 50, 60, 80. Nobel laureate has some Nobel laureate has around 100 or more than 100. Their impact factor, the H index. So institute has its H index, but it depends on the type of publications. So we have more than 200 H index, 207 at the moment, but every day it is increasing. So university is so big, every day some paper or the other is being published, and accordingly it is being added. So it is fast growing. So this is and this there are at least just to give a glimpse of these things, that there are not only how it comes that such a research and innovation, because there are at any point of time we have more than 500 around projects funded by the all frontier funding agencies like DBT, DST, NRF, ICMR, and IRI. So like that, there are also some international projects are there. So the theme of the workshop perfectly mirrors the research vision and of the university, and moving from the laboratory, we call it as a bench to bedside. So what we do in the laboratory bench, we always look forward that my research or our research is translated to the bedside, if it is a biomedical research or such research. Generally, it is for the society or the human benefit. It is ultimately user or the society or the human beings. So it is all to the, so that is bench to bedside. So the medical advancement requires the several disciplines by engineering targeted carriers to precise drug leading to the combining them with the AI-enabled the biosensor design. We are trying to save the future of the precision healthcare. So how it was earlier only the biosensor, but it was now we can think of that we can add the AI. So even the drug delivery also it can be added with the AI. So these are the major importance of this workshop or the new disciplines or the new science has come, new approach that it will again and again it will go change and change, and the science is always changing. So we have to look forward. We have started with the, for example, I was basically when I did PhD from BHU, I was a simple cytogeneticist or a geneticist. So that was the chromosome preparations and all these. So, but then it has gone to the molecular level, and we have learned after that, because if you do the same thing for after 30 years, 40 years, 50 years, then you are backdated. So science is always changing, and you have to adapt to the new development and new science discovery. But I will not take much time, and I once again welcome all the faculty and the invited speakers and all the participants to have a intensive learning, so that we have a hands-on training as well as lectures. So both knowledge and the practice. So I wish all a very successful and enlightening workshop. And I think also all the organizing members, team members of the ACNM, the Committee Center for Nanomedicine, who have designed this the workshop, excellent design, and the invited a lot of good speakers. And so this workshop, I wish all the best, and to creating the manpower in the field of drug delivery and diagnostic approach with biosensor, with the AI-enabled biosensor. So I once again thank you and wish you all the best for this workshop. Thank you very much.
Thank you, sir, for your insightful and thought-provoking address on this workshop. Your vast experience and visionary guidance have greatly enriched this gathering and will certainly leave a lasting impact on all the participants and young researchers present here today. With this, we now embark upon the scientific sessions of the workshop, promising an inspiring journey of innovation, knowledge exchange, and groundbreaking ideas. May these sessions ignite meaningful discussions, foster collaboration, and enrich us with insights that shape the future of science and research. It is my immense pleasure and great honor to introduce and welcome our esteemed keynote speaker for today's session, Professor Dr. Thirumurthy Velpandian, a fellow of the National Academy of Medical Sciences and professor and in charge of Ocular Pharmacology and Pharmacy Division, Dr. Rajendra Prasad Center for Ophthalmic Sciences, AIIMS, New Delhi. Professor Velpandian is a distinguished ocular pharmacologist and an internationally acclaimed researcher whose pioneering research contributions in ocular drug discovery, ocular pharmacokinetics, AI-driven QSPR studies, and innovative medical technologies have significantly advanced the field of ophthalmic sciences. He has mentored more than 75 scholars, authored over 225 research publications with over 6,000 citations and 12 patents in the new drug discovery, including a US patent, and developed numerous innovative technologies in drug discovery and medical research. His remarkable scientific contributions have earned him several recognitions, including Fellowship of the National Academy of Medical Sciences, FAMS, AIMS Excellence Research Award, Dr. Vinod Kumar Bhargav Award, and a Commendation Award by AIMS for the Newer Innovation Natasol 2025. He's also a recipient of the Sri Ram Deen Gupta Oration 2025 by ASEAN, Senior Joanna Memorial Oration 2024 by St. Joseph College of Pharmacy, Professor Venkat N. Reddy Oration by ARVO India, Chapter 2019, a Special COVID Award to honor COVID control by Ophthalmic Research Association 2021, and Grand Challenges Award by Bill and Melinda Gates Foundation and BBT, and recognition among top 2% scientists in the Stanford Xavier's Global Ranking for 2024 and 2025. We are truly privileged to have such an eminent scientist and visionary researcher with us today. Sir, may I please requestfully invite you to deliver your scientific talk and enlighten the audience.
Connections get more use. Connections that are used more grow stronger and more permanent. Meanwhile, connections that
A
are used less
handrail is not
fade
usually
away.
thought
Respected dignitaries of the dais, my faculty colleagues, and scholars, those who are attending today's workshop, namaskar. So today, the job of making the pitch ready for 3 days match has been given to me. Of course, you must say this workshop is a very unique one. So it has 3 different components. Well, and the bottom line is something which is AI driven. So everything is getting transformed into AI. So the time for us also to get into it. And now there is the buzzword everywhere is AI, right from PM to all the funding agencies now getting into AI. And it's a great opportunity. So I'm going to cover all these areas, but I wanted to keep my introductory talk very much that in a lower level that you can understand about it so that you can accommodate all those talks coming tomorrow, day after tomorrow. So my topic of today is biointelligence in pharmaceuticals, drug discovery to community. Finally, whatever you do, it has a presence for public. So it all starts from that side. Well, today, there's a very interesting picture. Once we have seen those films of Braveheart, where a man is fighting with a tiger, the next comes to play with the pet. Now, tiger has become a pet, but in front of AI, now you are so small, the AI is growing up. So when it comes to artificial intelligence, when you're just trying to understand what is this artificial intelligence in a layman language, when you try to say, a field of science and engineering concerned with the computational understanding of what is commonly called intelligent behavior. And with the creation of artifacts, and that exhibit such behavior. Now, there are many things here that artifacts exhibit such behavior. So you're trying to create something that is with the sixth sense. You know, we lit a ceremonial lamp. You know why we lit a ceremonial lamp? Every time you go to any function, The little lamp. The lamp is having 5 wicks. This represents 5 senses of yours, right? This is in Sanskrit called Sapta Sparsha Rupa Rasa Gandha. 5 senses. Through these 5 senses, you imagine this world. You realize this world. When 5 people lit the flame, all flames are same. The light comes out of it. Only things are different. So through this program of workshop or a conference, what we try to convey is people are different, but what they are teaching is same. That is a gyan, which comes out of the flame. That is what illustrated as a ceremonial lamp. But when it comes to artificial intelligence, what is the difference between human intelligence and artificial intelligence? Artificial intelligence, for this, you need to go to standard in school, lower standards, how many senses you have. So we always call, okay, 5 senses, but the human is always called the 6th sense. I'm asking this question to you. Which is the 6th sense? 5 senses are here. Which is the 6th sense? Right? And this is a gyan. Without that, you won't be able to recognize something through your eyes. So eyes are there, you are able to see something. Very interesting thing happened in ophthalmology. Somebody who is completely cornea blind, right? Cornea blind, or the lens is having cataract from birth, never seen the world. See, what happened is that such type of people exist in our country. Suddenly, a cardiologist, or ophthalmologist came, he got an opportunity to give vision by removing that lens, putting an artificial lens. Suddenly, the world is appearing. What do you think the perception would be? What do you think the perception of the person who has not seen anything? Now, he has been told he heard only through ears. That was a miraculous presentation. I really enjoyed. That baby could not recognize a round and a square because you need eye for your perception. The dimension happens somewhere else in your mind, right? So something in the 6th dimension is making that possible, that you call man or manas or mind, the 6th sense. What is well developed in somebody through which you will be able to interpret the intelligence and you try to understand. Now, when you try to create, always this question comes: can a robo or a humanoid be more intelligent than human? Impossible. Because your brain, if it is properly activated, if you are keep on looking at the mobile phone and watching Instagram, this will not be activated. It has to be activated by more and more knowledge. Adding more and more knowledge get into your buddhi, which is not there in English, okay? So somewhere it is getting recorded, and the intelligence of you is trying to interpret it. When you are trying to artificially develop this intelligence, you have to teach the baby how to recognize things. And that is where AI is getting into picture. Now, I'll tell you one by one, so you'll be able to understand this very nicely. See, first thing which has happened is the Deep Blue. This has happened, suppose 1997, one of the, our time famous Gary Kasparov was beaten by the brute force of computer, IBM supercomputer. He lost with the computer. First thing what we got is 1997. Well, this striked in the news and Nature, everywhere it reflected. Next came, little later, about 2005, DeepMind. AlphaGo has been, there's a next one. And this guy has beaten by DeepMind. So now he's a champion, a Chinese champion to do AlphaGo. So to play a game. So he has gone. So after that, is slowly. Now, the behavior adaptation has happened in the artificial intelligence. But how this artificial intelligence adaptation happens, and how, suppose tomorrow you wanted to train, suppose you are good in biosensors. Biosensor is a technique. You got a biosensor made. That's a complete old philosophy. You take out the old journal and you will get something called a banana electrode for detection of glucose. If those who already know about it, they know when I was a student, we were so excited. Wow, that's glucose oxidase is present in banana. You can use banana electrode for sensing sugar. So I'm talking about 1995. Even today, the technology of sensors has evolved, but still there are fundamental things are relying on. Either it is antibody mediated, or it could be a siRNA mediated, aptamer mediated, or it could be or nanoparticle bound, any binding agents you need it to make this electrochemical detection possible or visible detection possible. That's all about biosensors. But the word comes here is artificial intelligence. How do you train that? Now, electrode is one side. It gives you a potential difference. How do you try to train it to make something possible? So today, about 6 months back, somebody came with a technology, very interesting technology. And ICMR, we got that application. I happened to be one of the member. They submitted something very interesting. Can an infrared interference photometry, can it detect HbA1c? That's a very big thing. This is a challenge. Somebody said that I can detect, say all of you are having that the smartwatch, which can monitor your pulses, and you can, it's IR interference based. So based upon that, you are able to detect hemoglobin and carboxyhemoglobin, hemoglobin oxygenated or deoxygenated hemoglobin, you are able to sense it. But how do we sense amount of glucose there in that? So the interference, is it possible or not? So somebody has trained it, and he brought a possibility of it. So that's very interesting for us to see. Well, HbA1c can be picked up by an IR interferometer. That's very interesting. So that's a dimension which is going on. This, I think, most of you know about it. You keep your mobile on and keep the internet on, and you keep talking that, your friend is going to London. Okay. And you start getting advertisement of tickets for London. I think all of you seen it, right? So your Google is continuously hearing you. So what it is doing, it is sending somewhere. What it is getting converted, that is getting converted into knowledge. Who is going to use it? You will not believe there are big agencies funding money to figure it out. Today you sit, I even tell my students, do not go and search your innovation on the Google. I mean, anything, whatever you do, you put it in the Google and search it. When you search it, somewhere it is documented. After it is documented, it's no more your idea. Even before you come with an idea, somebody else is going to file IP for it. Today, IP war. I'm very happy to hear that, you have 1,500 IP, and many of them are commercialized. We're always proud of it. Congratulate Amity on this day of Technology Day. That's very interesting. I'm also coming here to speak on Technology Day, and I wish all Amity faculty and students for their success. So the moment you're, unless you switched off your Google, the hearing option, you say, okay, Google, the Google will start listening to you. Already it is listening to you, right? So everywhere you are being spied all the time, and the data is getting transferred, right? Yesterday, I found that Springer Nature has sued Google for stealing the published manuscripts as abstracts and converting them into LLM models. Yesterday, a case has been filed. So it's very interesting now. So whose property is what? If I publish a paper, it is my property. It is not your property. You already gave the right to somebody. So who is having the right? Nature Publishing Group is having the right, or somebody, Springer Nature is having, or Billy is having, or somebody else is having the rights. So today, and that they filed a case against Google for LLM model, right? Large language models. So today, LLM is one of the buzzwords, right? Today, we try to do a lot of things by using LLM ChatGPT. Right from Sophia, or it could be Amica, or it is, you call Chole or Hole. I don't know. Okay. That's another female. All are females. Okay. So they always made humanoid robots as female because that's the best representation they could have for the intelligence, right? And that's very interesting. So now humanoid robots have been performed in such a way that to even ballet. And I think all of you must be remembering about few days back, the half marathon in which I think last week, sometime like that. So the humanoid robot first win the race and has beaten the marathon runners. So that's a way how things are happening, and that's a way. See, this is very interesting. I thought I should show you this. Netflix has a different type of advertisement for the same film. It depends upon you. Already the Netflix always evaluate your character, what type of film you watch. Based upon it, it profiled you. The advertisement which comes up in that is always, suppose you are interested in somewhere, sort of a very adventurous film. So the scene will be coming into you is something looks like adventurous. Or depends upon your profiling which is being done in the Netflix, the advertisement goes. Can you believe? In Africa, like father is coming this way, and this is a way how it comes in Africa. The same thing comes in Caucasian countries, something different. There is a race bias, right? I mean, it shows the belongingness for a race. See how beautifully it is trying to encash your emotions. So now that's something that a flip side of it. You see, oh my God, fully from pharma industry to everything. Now humanoids have been engaged, whether it is the sensors or the hand or the sensors are somewhere else. Now it's so beautiful that packaging industry is being occupied by humanoids, and in pharmacy. In Dubai, I mean, recently the pharmacy has completely automated with robotics, and you will not believe. If you come to AIIMS, you see a couple of buses standing there with the robotic instruments. Now, everywhere you go, they ask you these two things. You want normal surgery or a robotic surgery, right? Robotic surgery, okay, I opt for robotic surgery. You pay some extra money. That is for the intelligence. Sometimes intelligence, you are paying only for the intelligence, not for the instrument. So that is attributed to intellectual property, right? It is an intangible asset. So now see the final thing, what had happened is that this is your remote, right? And you see the button here, Netflix button. Have you ever thought that why Netflix button is in my remote? Right? So you see what has gone. You purchased a TV and you purchased a remote, but that is having Netflix button. Why? That is what the whole business is all about. You will never come to know how a business is utilizing AI at any level, right? So your understanding is something like, before you engage in something, you should understand what type of AI is going on in that. Then only you win it. Otherwise, you are a victim. Only two things. So different thing, something like this is an AI representation of a film. How Netflix documents, you see that thriller, action, or is a parody or a romantic comedy. What type of liking you have, it profiled already. Now your advertisements will come so that you spend all your golden time sitting and just flipping through Netflix. And that is what it wants. The most expensive thing in life right now is your time. Okay, and they know how to make money out of it. That's what somebody is going to make money out of it. That is how the transformation has happened. So artificial intelligence, when it comes to pharmaceutical business, right? Drug discovery, pharmaceutical business, today is a huge business. And in this business, they only dictate what he's supposed to do, what he's supposed to buy, who's supposed to prescribe. To such an extent, they are deciding. So now hospitals are by corporates, right? So no, I know this doctor, I know that doctor, nothing works. Go and meet the CEO. He is a business guy. So you only see, oh, well, how much percentage discount you want? Do you have insurance? So everything is in the insurance, which we don't want is that. And insurance comes with your genetic card. Say that, okay, well, you have this many propensity for this many disease. So your premium is very high. This is the only thing which we don't want, and it might happen along with time. So now, what is important in whole scenario is, first is data gathering. So the Netflix is gathering data from the films what you have watched. This is a simple way to explain you. So it has gathered all the data, what type of films you have seen over last 1 month. Now Netflix gathers one more data, where you stopped and replayed. That is very important for them. Wherever you stop and replay, it is going to be taken a different way, because constantly you're using a digital input. So the feedback goes, and now you have decoders at your home. So decoders are two-way. It communicates also. It sends you. So now it communicates and gives the data. Now you become a data. Whatever you watch become a data. and the data is clustering, and now that bifurcates into different divisions, and he understands that what is your liking. So this is done basically data generation, and after that application of AI. And next is how is he going to use on you is the third question. So always these parts parallelly come. Only two things possible. One is supervised learning and unsupervised learning. Largely you divide the whole AI into two ways. One is supervised and unsupervised. Supervised is always you look forward for a data. Whether you use a biosensor or you use a biological readout or you use any information. Information is gathered and it has been segregated and it has been fed. So it comes as a truck. Completely, it is a packed data of different domains. So you are going to train the model, right? So everywhere, suppose for example, the time in next 2-3 months, now you stay, there will be a heavy downpour of rain in month of July, August. You'll have a heavy rain. What about drugs will be sold that time? What people usually suffer from? You'll be having a running nose. You'll be having upper respiratory tract infection. So the drugs, which type of drugs will go up in sales? This is for our simple knowledge what we have. So something like cetirizine, something like paracetamol, something like these things will be sold. All OTC products will be sold. Now pharma companies are ready with AI, and that AI is going to predict which month, which sales is going to be high, so that can be targeted, right? So now that's a business model. So selection of clinical trials, right from process optimization, everywhere you get into a manufacturing unit, there are multiple stages of manufacturing unit. Everything is automated. So it's all again AI control. AI even decides. Suppose, if you're in a pharma industry, you are going to run a big reactor, the bioreactor, in which you are having a feed of, you are going to do fermentation process, and you're going to feed with some intermediate. How much amount of intermediate is there? What type of segregates are getting formed? If you make an AI, you know what will be the ideal situation. At a particular condition, sodium bicarbonate level is going down. You don't have to go and just take it up and do it. Automatically, it feeds the bicarbonate. So the pH is rising up. So the optimum conditions are reaching, neutralizing lactic acid formation. So fermentation process is optimized by AI because there is a readout and there is an optimizing AI. So chemical synthesis days are gone. We're gone for, today is microwave synthesis as well as parallel synthesis. It's gone to second, next level that you can do for several compounds can be synthesized immediately in a multiplexing manner. Similarly, that understanding a pathway. So many of you are interested in omics. This could be proteomics, this is genomics, transcriptomics, and you have got, lipidomics, glycomics, all omics platform. You merge it together. You wanted to get a centerline hypothesis. Most difficult one. Looks very easy. Always student comes to me and they say that, the young investigators come to me, say that, sir, I am going to search for something in this as untargeted proteomics, untargeted metabolomics. I'll figure out which is high in the disease. So I'm going to see whether it is there in the control versus disease population or not. I'm telling you, the moment I give the data, you will come to know what is the problem. It's a heap of data. It's like searching for a needle in the heap of data. It's very difficult. People, those who already worked into it, know very well about it. It's very, very intensive. Now, again, here, you try to use artificial intelligence so that can be possible. So without understanding this, you cannot get into omics studies. So to simplify the data, digital transformation in pharmaceuticals everywhere, distribution, inventory management, regulatory compliance, clinical trials. Clinical trial is one of the very large time-taking one, in which again, now AI. Today, when we handle, I'm in the ethics committee of institute, so we get lot of applications. Now, today, earlier, we used to get all physical submissions. Today, we get everything online. And when the moment online comes, hereafter, I'm very sure in another few years, we know that there is a small AI is set up there and itself decides what is what is there in the application, what is not there in the application, which section to look into it, which section not looked into it. That much of automation is possible even in that itself. It's very easy nowadays. So personalized medicine, we always look for optimizing some things, what we call repurposing. So we have got enough amount of drugs, we are having compounds. Repurposing is something that, again, much talked about. So already drug is there, already proved in many hypotheses. So it is always try to look for it for a new target, which is, again, it's an easy one. So everywhere, right from e-commerce, healthcare, manufacturing, transport, finance, banking, everywhere. The steps in drug discovery, right, from the preclinical studies to phase 4 or phase 3 studies, it takes 10 to 12 years. But today, when you see the fast tracking, you will not believe recently a drug has been approved, orphan drug, very fast. It's the fastest artificial intelligence made drug. So every section, like synthesis to biomarker, generating data models, repurposing of extenders, everywhere it is coming up. All AI, along with robot, now become humanoids, are going to replace many places. I don't know how much it is going to replace human takeover, which we don't know. Along with the time, we'll come to know. Let me just get into a small problem, what we had and how we optimized it. This would be a practical understanding how we dealt it. So what is something very interesting is we always publish positive results, something very unique. Anybody publish and they publish only positive. And you will not believe, you take one herb, Indian herb, it will be having multiple properties. You take tulsi, right? And tulsi or neem, you see number of publications came from that. There will be a whole lot of publications, all character, whatever you think of, it will be there. Now tell me whether something wrong with the compound or something wrong with us. How can one compound have multiple properties? So now we got a new terminology in pharmacology that are called adaptogens. They adapt to what you want. आपका क्या चाहिए हो जाएगा, कोई बात नहीं. There is a Hindi, there's a beautiful word, कोई बात नहीं. The person who came very fast and he hit me. Okay. Then he said, oh, I fell down. He said, you should suppose say, I'm sorry. He said, koi baat nahi. Very nice. He said, koi baat nahi. So, theek ho gaya. Koi baat nahi. Don't talk anything about it. Uska literal meaning, koi baat nahi. We are not going to talk about it. You got injury. I don't want to talk about it. How nice we are, right? So that's how it is. So many a times, negative results we don't publish, right? So now what happened as a result? We got several compounds. For example, terfenadine. You remember that in 2000, sometime in 1995, 96, this drug came as a blockbuster drug, terfenadine. Those days, when I was a student, when we were running nose, the first drug ever, non-sedating antihistamine. After that, citizen came. First came is terfenadine. The terfenadine caused one severe side effect, caused disappointed arrhythmia for selective patients. So the company, what it did, slowly they worked upon, it took its metabolite as fexofenadine and they went back to FDA, said, look, terfenadine, my molecule is having problem. So I'm coming with fexofenadine. It doesn't have that, our own body makes it, okay, fexofenadine. That came up with a drug that market is even today in market. So similarly, there are many drugs which came with side effects, has completely killed the drug. These are only some success stories, but negative stories, we never talk about it. Similarly, there is one incident which has also happened. So memantine is an excellent compound, came for neuroprotection, for Alzheimer's, Parkinson, Huntington, ALS, that is atrial lateral sclerosis, vascular dementia, CNS trauma, as well as cerebral ischemia. All these places, you have something in common, that is excitotoxicity. Excitotoxicity by glutamate is a central line hypothesis for all these things. So for Alzheimer's, today Alzheimer, if you find a drug for Alzheimer, is $1 billion business, right? Because, I mean, a lot of aging population in US and other places are all suffering from Alzheimer's. So if a drug, if you get it, it's great. So US FDA approved it for Alzheimer's because they found that it is blocking this excitatory amino acid transporter. Thereby, it is cooling down this excitatory amino acid pathway so that The quick confirmation is Alzheimer's is somewhere in the brain. So no, the blood is having a barrier to reach the brain. That is called a blood-brain barrier. What is this barrier? It is like Amity's gate number 1 or gate number 4. Show me your ID card. No. Show me your car number. Oh, this number cannot go inside. It's not like that. That it doesn't have a guard standing there to look into your ID, right? So how is this barrier functioning? We don't know. We don't know anything about this barrier. Now that is blood-brain barrier. So similarly, we know there is another ratio. There is something called blood-ocular barrier. There is something called blood-testicular barrier. There's called blood-ovarian barrier. All places where nature wanted to protect your valuable organs, the barrier is present over there. What is this barrier? Is it a physical barrier or a chemical barrier? Or what is this barrier? Something which went on, very interesting thing about it. So what, to cut short this guys, what they have done it, they found the memantine is having great effect for Alzheimer's when it has been approved for Alzheimer's. So they said, well, why shouldn't we try this for glaucoma? Glaucoma is one of the progressive disease of the retina, and the retina goes completely bad along with time, and that is also due to excitotoxicity. We always consider retina as an extension of brain, because you see the optic nerve comes from brain directly. And once if it goes on, that's all. Nothing can be done. It's not like peripheral nervous system. So the company wanted to trial, and they did the trial for 4 years, giving the same dose what you are giving for Alzheimer's. Simple hypothesis. If it crosses brain, it can cross eye also. So simple logic, because brain and eye all same, so it's okay. And then this publication came calling that the moment the day of the music is died after cupping up over $115 million in 2008. So Alagan lost this game, right? So completely gone. The trials failed to show any small. So what happened is that then there was one article came in drugs news, the drugs R&D. So editor asked me to comment on that. So that day I opened up this case of memantine. So, as being, we always work upon pharmacokinetics and ocular pharmacokinetics. Therefore, our interest went on understanding, is it capable of reaching the eye or not? So we went it out and we did a lot of calculations, and we found the drug is not capable of reaching there. You need 1 micromolar concentration to have a neuroprotection. It hardly reaches 0.3 micromolar. Therefore, it could not have any protection. The drug failed. What this company should have done, they should have conducted that studies to ensure that it is going inside. So today, suppose if any eye injury happens, right? Today, when I joined in my center, that time, hardly I used to see any diabetic neuropathy or diabetic retinopathy patients. Today, every week we are getting hundreds of patients. 1 out of 4 among us is having diabetes. And down the line, and in 20 years, I think 1 out of 4, 90% of us are going to have eye problem. This is that statistic says, or kidney problem, or you are going to have a myocardial problem. So if this is going to be neuropathy, retinopathy, nephropathy, any one pathy will catch hold of you. So this is what it is. Now, have you ever thought why so? Why we have diabetes, this high diabetes? One of the American presidents said, Indians are eating a lot of rice. Is it so? Right? So we really don't know. Our parents lived they lived long. They never had it. Our forefathers never had it. Why now? Young females, out of 10, 40%, out of 100, 40 of them are having PCOS. Yesterday I came to know they renamed PCOS as metabolic syndrome. PMS, they changed it into it, right? So the PCOS has become a bigger issue, something which is common for all, that is called insulin resistance. Now you answered the question, why insulin resistance? What is causing insulin resistance? Suddenly I am gaining weight. Despite I eat very less food. Why? But everything is fine, but somebody is getting PCOS. Why? Something which is having endocrine disruptor, which really we don't know. These questions we need to answer because our own societal questions, right? So similarly, The drug doesn't go inside the eye. Suppose if somebody is having diabetes and I want to protect the eye, so I may like to give a drug orally. If somebody has an accident or somebody has an infection, I cannot give an antibiotic because antibiotic won't reach the eye. Antibiotic can reach anywhere in the body, cannot go inside the eye. So this is my first problem when I encountered when I went to RP Center. So ever since, so much of emphasis has been given for Alzheimer's and all the things by using AI. So we thought, okay, let us try to work it on. How can we handle it? That it came very handy about AI. So that's what I'm going to just give a glimpse of it. So this is a beautiful structure of eye. You say posteriorly, you just take the retina, and retina you divide into two types. That is, these are all retinal layers, multiple layers of retina. These are photoreceptors. There are choriocapillaries, and you are having a blood. The retina is having its own blood supply. That is we call an inner blood retinal barrier, and the choriocapillaries we call outer blood retinal barrier. The cornea is another big barrier. It doesn't allow any drug to go inside. That is why when it comes to eye, we use 0.3% of fluoroquinolone of ciprofloxacin eye drop. And when it comes to dilating your eye, those who are wearing specs, they know when they undergone testing, they must have put a drug and it's all dilated, very high concentration drug. They use it. Why? The cornea won't allow things to go inside. If so, you take a bath in a pond, all toxins will go inside. So the well-protected barriers are everywhere. Then how to breach the barrier? You want to make the drug to go inside, right? And it is immunoprivileged. There's no blood supply inside vitreous. While the surgeon is doing cataract surgery or vitreal surgery, the biggest fear of them is the patient should not get into endophthalmitis. That is called infection of the eye. The patient will lose the eye. You see, often you read in the newspapers that a cluster endophthalmitis, people have gone blind. That's a dimension. Unfortunately, all of us at one day has to undergo cataract surgery. You understand? See the dimension. So you are in the hands of somebody, the doctor will ask you sign the consent form. Even if I go blind, I will not sue the doctor. So he's okay. So now you are in trouble. So how to save people? Every doctor wants, no, I want my practice to go well. I don't want patient to suffer, obviously, right? But he cannot treat the patient with an antibiotic because the whole world of medical world is trusting on antibiotics. A patient comes, a worst road accident victim, come to trauma center of ours, we treat them nicely. Beyond us, we always, concentrate on antibiotics because they save them, right? They don't get infection. But today, when it comes to eye, we are lagging behind. So we went on understanding about something. Multiple studies have been done. To cut short, few studies, which we took the help of DRDO. In this case, IMAS was Dr. Asim Bhatnagar was a close friend of mine. So we did this gamma camera studies, an excellent proof to prove that the barriers are working. So what you see here is a rabbit, and you see this balls, two big balls of white glowing balls. When we inject, this ofloxacin labeled, technetium labeled ofloxacin, we inject into the eye. After some time, within an hour, you see the bladder. The bladder is completely bright. And you see the kidney, you can see the stomach, everywhere, the glowing. So it's going out of eye and it's getting excreted through the kidney. But what we did is, we used a blocker. Then we know that. Now, no more, they are simple barriers. They are much more sophisticated. Very typical, what we call drug transporters. They're called xenobiotic transporters. These transporters are cationic transporters, anionic transporter, peptide transporter, polypeptide transporter, oleonuclear transporter. There are many transporters. They decide what to go, what not to go. It's not that the once if peptide is having an identity of peptide is allowed, otherwise it is not. Now you go understand these barriers. The barriers are it's just a molecular tunnel. The molecular tunnels are functioning based on electrostatic forces. Something is positively charged, it is pulled in, or negative charge is pulled in, it's again charge mediated. Somewhere you see now from the dimension of biology, you've now gone into physics, something into quantum, something into molecular energy. So now the transformation is happening from biology to another level. fundamentally, when you are looking into the transporters or energy ports, and the energy ports sometimes utilize energy, sometimes they could be co-current or counter-current. I don't want to go more in detail, but a multiple mechanism. When we stop the functional barrier this here by using a blocker, now you see the bladder here. At the same time, you see the bladder here. See the difference between the stark difference between these two. The first time showed us, wow, it is true. What we thought is right. It's much more sophisticated. Then we went on understanding which type of drug go in, which type of one will not go. The transported mediated transmission of drugs. So finally we understood something that why, what's happening? So it's something like when you inject the drug directly, it's just getting pumped out. But when we give IV or through systemically, it is trying to go in, but the pump is working in opposite direction. So it throws out. So this is the first time we understood that the involvement of PGP efflux transporters in the eye, how they are governing pharmacokinetics of the drugs. So for all these studies, all these studies came from our lab to prove this hypothesis first time. So ever since we have been working on several transporters, that is on nucleoside transporters, organic cationic transporters, then we understood several transporters are there. And targeting transporters is the best way to reach a particular organ once if it is guarded by ocular barriers. So this is so we finally found that something like cationic in nature, which is positively charged in nature, is capable of taking in and get excreted through, another slow way of elimination. Therefore, it is reached very nicely. So similarly, what happens when the eye is getting into inflammation? Every ophthalmologist said, once if inflammation happens in the eye, your barriers are lost. But when we just gone into a particular experiment to prove, no, we did not see that way. A particular transporters are getting upregulated even before entire destruction comes to the complete eyeball. So before that, again, it doesn't allow some of the drugs to go in, and out of which the main drugs what we use for inflammation is steroid. They themselves are not able to go inside. Therefore, you cannot handle anything except you see that eye is going bad if somebody. So there are a lot of molecular studies we did, and we just inside an understanding about it. Then we came about biointelligence. This is very interesting. What we did is we took up, I think next slide I show you. So this is the final concept we developed out using artificial intelligence again. This is using a machine learning, algorithm, in which what we put the hypothesis is that when you put a drug or a drop in the cornea, that is in the outside eye, the drug is getting ionized. So now the pH of the precorneal area is 7.4. You put a drop of a drug, for example, you put ciprofloxacin hydrochloride. The ciprofloxacin hydrochloride is having typical pKa of 4.4 or 2 to 3.2. But it is 2 pKas, but that pKa, at that time, fundamental Anderson equation explains how much is ionized, how much is unionized. See, now you see slowly we are transforming our idea into a little more on physics towards quantum mechanics. Now, already I told you what will be the parameter which could predict that transition, electronic transition, and on the energy pathway called transporters. We found that there is some beautiful prediction in the quantum parameter that is called gap value. That is between HOMO and LUMO. Highest occupied orbital and lowest energy occupied orbital, LUMO and HOMO levels. In between that, the gap is called as gap, gap, the energy gap. So that is molecularly very, very capable of predicting what can go, what is not going. So at the same time, with this equation, using PSA, there is polar surface area. Another one is its log P value. Another one is pKa of the molecule at that particular and the gap of that particular compound. We could predict beautifully which type of eye drops can go in, which cannot go in. To predict this, we have done a beautiful study. So to do such a type of studies, so broadly you divide AI into two types, right? One is, unsupervised learning methods like ANN, CNN, etc. Another one is machine learning, AI, ML. Always it's called AI, ML, artificial intelligence and machine learning. The machine learning hypothesis, we took it here, in which the supervised ML is again, we try to create an algorithm, and that algorithm is going to predict. How algorithms are getting created? The hypothesis is what I told you. This is what we expected, and this is what we observed. Okay, sometimes we try to make a hypothesis and work upon it. Sometimes we try to explain what is happening over there. So based upon multiple parameters, you try to mix and match to reach which is the best parameter to go inside. And that is how we try to do it. In unsupervised ML, you have clustering of the k-means clustering, hierarchical clustering, DB-clan clustering. There are several models are there. This is a small thing which you need to learn how to apply, when to apply, which model to which type of problem. But remember, first is data. Another one is predicted its property. It could be a biological property, or it could be a biosensor's readout property. Anything it is. In biological property, it could be anything where what the drug or chemical is going to do into that. Today, sitting with the AI, you will be able to figure it out which type of drugs can cause Trosidase D-point, the one which I told you about tafenadine. Similar type of compounds. So we call that as a negative screen. You remove right from the development part. Something which is going to interact with PGP, remove from the development. These are all the drugs are not going to fetch us anything. Later, when we come to clinical trials, they will fail. So you try to use prediction models, the beginning itself. Creating prediction model is in your hands. You can predict anything, but the world, the limit is sky. Sky is the limit. So your idea is something which is going to transform these things. I hope you understand what am I talking about, right? So this is how you do it. So, I mean, don't get so much bogged up with, oh, yeah, it's not my cup of tea, something. It's very simple. There is now today, everything available online. There is something called Anaconda browser. Okay. Anaconda browser. It's very interesting. The Anaconda browser, which is having Conda, and you just put it, you get NumPy, you get lot of free softwares. Then, you know, you really jump and joy seeing that kind of interface. So it's easy for you to learn and go to YouTube, right? Learn about how to use it. So many people freely teach you how to use it and calculate it. This is a golden way so that you can understand. So how supervised learning is going to transform. You see, this is a way how somebody has designed a software. That software is going to diagnose your disease. That's called an ADA. This software, this is an app. You download the app and you can use it. It will ask you questions. Okay, you are not well. So what happened to you? You're having headache. So do you have fever with headache or simple headache? You say, I have fever with headache. So if fever with headache, your nose is running or you are having, so you keep on asking questions. You keep on answering the question. Finally, you might suffer from this. You better visit a consultant. So many people, what they do now is, my colleagues say, Dr. Saab, ऐसा दवाई लिखी है जिसको जो साइड इफेक्ट ना हो. Okay, देखो साइड इफेक्ट ना हो. There is an effect, then there's a side effect. Can you name something without side effect? भाई 2 लीटर पानी पी के देखो, साइड इफेक्ट बताता हूँ. Okay, so this is what it is. थोड़ा सा 2-3 चम्मच आप नमक खाओ, साइड इफेक्ट दिखाता हूँ. So this is how it is. Nothing is safe in this world. When you handle everything in limits, that saves you. Right? So you should know, I gave you a beautiful knife to cut the apple and eat it. But the knife you should not use to cut your finger. Right? That's what it is, a double-edged sword. So this decision tree is another way to which you do the analysis. Whatever I am telling you, it is there in the Anaconda browser. You have so many. You go inside YouTube and learn. So you'll be able to easily learn how, okay, nearest neighbor. Suppose, for example, you're having multiple hostels. You know, Amity have multiple hostels. A new student is coming. Okay. So he has some, weight, height. For example, the hostel contractor says, I make only 20 kg of atta every day. I cannot go beyond this because my tolerance cannot hold it. Now another person says, I have 15 kg. Another person gets some other caliber. Now you want to distribute this student in somewhere. In such a type of scenario, he might be a vegetarian, non-vegetarian, or he could be a Buddhist, or he could be a Jain. So you have multiple, this one, right? So sometimes every hostel provide something on non-vegetarian, something on vegetarian. Now the AI will tell you where you have to put him, on which day where he is supposed to go. So even it customizes somebody's liking on the existing platform by using this K nearest neighbor can distribute it. It's all mathematics, Euclidean distance. When you just go inside, you'll really enjoy working on it. So all the equations, what we generated using multiple linear regression. And we gone into multiple linear decision tree regression or random forest regression. So use multiple types of regression can predict. Any type of curve can be predicted by using this and the relationship between the effect and the concentration. Concentration, I'm telling you in a more scientific way, but character versus effect, you can be able to predict it by using these algorithms. This is something Anaconda Navigator, right? So I told you about this browser. So do it, you have all of them here, NumPy, Matplotlib or Pandas. You can install anything and you can do every damn calculations, I mean, this is the first thing what I learned when I start working on it. Therefore, it is not something very, if it is easy for me, for you, it is nothing. So you can learn much better. So artificial neural networking, just see that. Something very interesting. I want to ask you, how does artificial neural networking work? See, in 1943, there's a guy called McCall and Pitts. One guy is a neurophysiologist, another guy is a mathematician. There is something called a neural summation. You need to understand the fundamental basics to understand what is this ANN. Okay? Artificial neural networking. Many people, even go to engineering, they talk ANN, but you are from biology. You should be able to say why, how, right? So what this guy has done is, always you should have collaboration. See that? One is a neurophysiologist, another one is a mathematician. What is his problem? Very simple. There is a neural summation. A neural network, what happens is, it gets the dendrites coming from different neurons, the same way how I shown you. This is somebody's beautiful picture how our brain neural network is existing. So it is something like one neuron is linked with another neuron. Sometimes multiple neurons are linked. So linked to what happens? What happens here? Just you see this.
During this important.
This one you see. Very interesting. So a positive charge is coming from multiple neurons or coming into another one in which commonly they are coming. This is called neural summation. They're all coming together. But what is happening is, if this gets positive, this neuron is getting positive energy from all the places. You have a pointer? So this is having all positive coming, but remember, the end of this dendrite, this synapse, what is going to do it? It is having GABA as a transmitter. The moment positive charge is coming, but what is getting released is, what is getting released is something. Oh, thank you, sir. So what is getting released here? Suppose GABA is a negative neurotransmitter. Everywhere positive is coming, but finally it is negative coming out. So that is how the neuron summation is doing everything and goes to the fundamental basics of and or NAND gates. You must have studied in higher secondary school, the gate mechanisms. And that is how the summation is happening, and that's how. So when we try to do artificial neural networking, we try to teach the baby how to do that. I have a small video to show you how we teach this baby. Okay, so this is neural networking about it. So that how she is teaching you say.
A handrail is not usually thought of as a tool, but it is. And baby humans come to depend on them just as adults do. At Karen Adolph's lab, researchers ran a number of experiments using a handrail. The first thing they wanted to know was whether a 16-month-old toddler knows when they need the support of a handrail. As the bridge gets narrower, Stephanie becomes more cautious. And when it's too narrow, she pats the ground with her foot. and then looks to the researcher for help. Then a full railing is put in place.
Get some Cheerios. Look. Get some more in the box. Look, honey.
Stephanie taps the handrail and makes sure it's solid.
So many in the cup. You can get some more. Come get the Cheerios. You can do it. Once she figures it out. Yeah. Come on.
Grasping the rail, she decides to trust it.
Good girl. Get it. Ring, 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 ring.
So this video shows how all of us learn the world. How we know how to walk, right? Every step, cautiously you do it, and you try to learn. Of course, your brain is getting trained. Your neural network is getting trained towards it. But now you developed something, you want to train it. Sometimes you have to iterate it 1 million times. Then only the neural network start working, right? So how you do all these things? See, the neural pruning happens along with time. So
During this important period of brain development, billions of brain cells called neurons send electrical signals to communicate with each other. These connections form circuits that become the basic foundation of brain architecture. Circuits and connections proliferate at a rapid pace and are reinforced through repeated use. Our experiences and environment dictate which circuits and connections get more use. Connections that are used more grow stronger and more permanent. Meanwhile, connections that are used less fade away through a normal process called pruning. Well-used circuits create lightning-fast pathways for neural signals to travel across regions of the brain. Simple circuits form first.
see, this is a neural pruning is happening in a young toddler. It's very important at that age to train the baby. Today, what is happening? So actually, a few days back, I was called in autism public interface to discuss about it. There I discussed about this. Today, we see a lot of things around, right? So the baby is crying, the mother gives mobile phone. Now the brilliant brain neural pruning happened. Seeing the mobile phone, what do you expect from the baby? Right? And the baby mother says, without watching this mobile, this baby is not eating. Right? I mean, when you are in a society, you're supposed to take the message, you should tell, don't do that, right? So that's how it's happening.
During
Neural
this
pruning happens, and that's how the neural summation happens. Artificial neural network happens like this. What do you call that is the rectified linear unit. ReLU, it work. When you just start working, understand, everything is converted and sent it to, it's all fundamental matrix. I think you must have used matrix in schools, right? The same matrix works, and the matrix is getting converted into a complete algorithm. I'll show you a small example. So this is a complete network. This is a first layer, and this is a signal. And the signal goes inside. When you start training the network, and this is what happened. See, first layer, again, the few layers are happening, the different shapes are coming, but finally it is reaching here. See, the question is whether it is a circle or a square or a triangle, I don't know. 4 questions. But here it is continuously coming. See that. Now it is coming here. It's a triangle. Yes, it's a triangle. Now you say, is it a triangle? No, it's not. I mean, it's not a triangle. The square. You continuously train them, then there is something which you cannot see. What is its path? It is getting created, but it links with the reality. That is what neural networking all about. Okay. So this is how it is. Suppose if you see a picture, how picture is getting converted. The picture gets converted to pixelated first. And the pixelated pictures, I hope all of you understand what is pixel. So now, see, the pixel is getting converted into a matrix. Now it is a matrix. There are a lot of dots. First, it is getting converted to black and white. And the color is getting converted into some number. So the gray scale has been created from 0.1 to 0.9. And the grade is being given. Now it has come as a number. Image is converted into number, and the number is fed into the matrix, and that matrix is going to use it. Suppose you must be using that fingerprint reader or a face reader. Your mobile phone face reader works by this way. How your face is getting converted, how it is getting linear conversion happens, and how is it getting linearized into matrix form, bring it to the ReLU, and how is it getting. So if it is instead of you, somebody picks up the phone, it says, no, I cannot open the phone because it's not owner. So that's how it works. Once if you start learning more, you understand more about how it works. So in AI, medicine is having a lot of bifurcations. Medicine, the machine learning is something very interesting where you look for data and you have a sensor and you have a readout. You try to do it, you convert that into an AI platform. Or deep learning, you don't know. One aspect you don't know. Use deep learning. It will make a pathway possible. Something like I am just getting the signal from IR sensor. I want to look it with the glucose. That is possible by using AI and deep learning. So the fuzzy logics always works to make it. And this is how that electronic state quantum parameters are working. This is how we converted in a lab about, we have used a technique called a cassette dosing technique. That means we give multiple compound to one animal. And since we had the power of mass spectrometer, so we take out the sample and analyze for all the drug concentration. So with single organism, multiple drugs, we will be able to do it. So similar type of it done to create that kind of data. And we went on understanding about it. Today we know this equation predict which type of drug can go inside eye, which cannot. So again, this quantum descriptors are now already I discussed this about it. This is about taking drug orally, which can reach the eye. This also we know about it by using the similar principle. Again, we used a rabbit to create it. The whole scenario is given here. The drugs congeneric or non-congeneric having similar structural analogy are called congeneric. Diverse molecules are called non-congeneric. So, similar molecules, we used it to feed it to the animal and take out the blood sample and the aqueous humor or a vitreous humor gone for doing all the analysis. Now we know how to predict it, which can go inside. That's what has happened over the period of time. So that's beautifully predict. Now the beauty is that we were able to predict even human penetration data, which is very difficult for us to do. So finally, we know this is a schematic representation, how we do it. We give by IV, it goes, or orally you give the drug reaches here, it's calculated, it reaches distributed in the body, it reaches the blood vessels, and how much it goes inside is defined by this equation log P and the gap as well as protein binding. The more amount of protein binding happen, less amount of drug available for action. That at the particular pKa, how much can go inside, what is the plasma level which can create and which can cause how much amount of vitreous levels. That we were able to predict it by using the single logic, by using ML equations. Now, it is not restricted only to this. The whole idea is given to you. And this is the way we predicted by using PCA plot, how we predict the metabolomics data and converted into a sizable way to understand about the trace amines. We found first time the trace amine involvement in the eye diseases. So this is again going on. So there are multiple papers. I'm not going to explain individual of them about how to use machine learning. This is for you to understand to design polymeric long-acting injectables, drug delivery systems. That is what the thing is about. These publications are available, in the PubMed. You can go through it. My purpose of my talk is giving you an idea. But your sky is the limit. You can always look for it. If you are having, a wine, it's still very interesting. Why a particular wine from a particular place is very tasty. Somebody did this work. I really like that. They just used this plot to say, what is the composition of that wine which tastes better? Can you understand this? Everybody loves a particular brand. Why? Now we know what is there in that. We can create it, or which one is original, which one is duplicate, you'll be able to identify.
QC has evolved.
Not going in detail about it. Lot of IA-based computer-assisted tools, they are free of cost. Okay, I don't look for it for I need a sophisticated software. I need this. People, those who are not intelligent, only say, I need a huge money to do research. What is required for the research is inquisitiveness and your eagerness to learn more. That's all. So the multiple uses in pharmaceuticals as well as drug repurposing and all. So with this, I come to you again, lot of publication. I want you to go through PubMed. There are things available. So AI is a double-edged sword, as I discussed to you. Biointelligence is transformation causing automation. It's up to you how you try to do it. ML models. I have shown you one example. Now you can try yourself. Training data, which is sets are essential for AI/ML. And data science is going to be the most admired, wanted professionals in the century. So I wish that, you take this lesson from this workshop to make something. I thank my students, for their contribution in many places in this. And thanks a lot for this opportunity to talk in front of you, especially Dr. Madhumika. Thank you very much.
Thank you, sir, for such an enlightening, engaging, interactive, and thought-provoking talk on biointelligence and pharmaceuticals. Your lecture brilliantly highlighted the transformative role of innovation, data-driven science, and interdisciplinary research in shaping the future of modern therapeutics and healthcare. So, now it is my honor to welcome Dr. Rajkumar Tulaswani, Scientist E, Biomedical Research, DPAS, DRDO, Ministry of Defense, as a speaker to the scientific event. So I would like to request Professor Seema Bhatnagar and Professor Madhumita Ghosh to welcome Dr. Rajkumar Tulaswani with the sapling. It is a moment of honor and privilege to introduce and welcome our distinguished invited speaker, Dr. Rajkumar Tulaswani. Dr. Tulaswani has an extensive expertise in biomedical sciences, including pharmacology, toxicology, cellular and molecular biology, as well as advanced analytical techniques. He also possesses significant research experience in handling and studying chemical warfare agents. His remarkable contributions have earned him several prestigious honors, including the National Award by the Seabuckthorne Association of India and the DPAS Laboratory Scientist of the Year Award by DRDO. He has successfully developed innovative products such as DIP GFIT and DIP CFIT, and adaptogen, herbal sanitizer, and herbal sanitizing wipes and towels, along with technologies related to high energy laser systems for both in vitro and in vivo exposure studies. Let us extend a grand and heartfelt welcome to Dr. Rajkumar Tulaswani as I respectfully invite him onto the stage to deliver his enlightening address.
Good afternoon, everyone. It's already lunchtime, so I will not take much time over here. Today, I will discuss some of the technologies on the targeted drug development and what are the barriers and how we can cross with the directed energy technologies. Directed energy technologies encompasses all the spectrum, what your mobile has, 2.4 gigahertz, what you see in your 5 gigahertz, what you see in your wireless communications, in defense systems. So whatever the waves and wavelengths and every spectrum, you have microwaves in your houses, they all encompass the energy sources. What is the difference? The difference is the systems which you are using in your houses or in appliances, they are low energy, diffuse, and non-coherent. But when we come and talk about directed energy technologies, now the physics has advanced a lot in last few decades. Now you have the optics to make it a coherent. You can make a wave in a very coherent way. So a larger wavelength can be coherent on the one same spot. A high energy is delivered. So this directed energy, the irony is I work on the defense warfare systems and I use for the warfare systems. And today I am talking about the targeted therapies. So this is contradiction. We are working for the warfare, but now today I will take you to the directed energies for the targeted delivery systems. How they are the future of the medicines and how they would be helping us to overcome all these barriers in the targeted drug delivery systems, which still we are facing in the scenarios and throughout the ages. this you will going through the most of the workshops. I'm just touching about in 1906, magic bullet was thought of. Elric thought that he will give some medicine, that medicine will go to the exact disease spot and will be released. And so he conceptualized in 1906. So what is the target drug delivery system is? You have to have your medicine to go to the sites like tumor sites, some sites. So it may help to reduce your systemic drug doses, and then it will reduce your cost. So targeted drug delivery systems are the precise requirements in the medicine. So these are some of the benefits. They avoid toxicity, adverse reactions because they go to the target. Encompass number of drugs and gene can be delivered to the specific sites. They can facilitate the site actions without disturbing the other neighboring tissues. And the therapeutic payloads directly go to the tissue target so that you do not disturb the other organs. So it may save your cost and give you the specific desired result. The targeted drug energy became very complex after the emerging of the blood-brain barriers, how the organs are protecting themselves, and the tumor cells. So now the cancer cells are making more problematic to us. So that's why the targeted drug development concepts came in. So already Dr. Pandyan has told there are a number of brain barriers. What is the problem with this blood-brain barriers? Every organ has their own mood swings. So they are behaving their, every organ has their own blood barriers and all have their own habits. Brain do not allow most of the molecules. Liver engulfs all the exogenous particles. Kidney eliminates everything. Liver has their own blood barriers. He becomes selective. So there are blood barrier is everywhere. So they are becoming like moody. They are moody. So now you have to bypass the system. So this is a challenge because now you have to target every organ. Now you are believing in a targeted drug delivery system, but you have to have the system. So lots of research is going all around liposomal nanomedicines, liposomes, siRNA delivery medicine systems. So there are n number of systems are going. Why? So much target drug delivery systems are being designed with different vesicles is because of the moods of these different organs. Having the similar identical single in vivo system, you have so much complexity with so much blood-brain barriers. So these blood-brain barriers encompass pharmacological barriers. There are pharmacological barriers. Some organs believe in their physiological clearance mechanism. So there are n number of mechanisms, pharmacological challenges for these drug developed systems. So that's why these challenges has to be overcome. Still, you have so many targeted drug delivery drugs, but still we have not received to any drug which has the 100% efficiency. So why? Because complexity in the systems. Within the in vivo system, there are more complexity within the organs. So some are having the pharmacological reasons, some drugs are going through the pharmacokinetics reasons, some are for the pharmacodynamic reasons. So these are some of the, again, problems at different stages. So there are, what are the difference between targeted and conventional drugs? Targeted drugs are more selective, have lesser side effects. They are believed to be more having efficacy because they are directly reaching toward the target. They can be used more clinically, judicially used, and maybe a cost effective. There are a number of ways to target the drug delivery. It may be active, passive, it may be a disease-based, or it may be a location-based. So passive is a natural process where you are targeting some drugs, they are going and they are locating their disease pathway and destroying. In active, you have the ligand-based, external stimuli-based drug therapies. So these are the different passive targeting, active targeting molecules. And some targeted drugs are based on their selectivity of the target because you have some tumor, you have some pH characteristics, some you have temperature, enzymatic activity. If you know, you can target them as well. So there are number of ways how you can target your diseased organ or tissue. There are number of drugs has been approved, but still a few drugs have been approved, but still the quality adjustive life years has been increased, but there is no overall outcome or the disease prevention has been come in the over years. So what I want to now discuss with you is that this is the targeted challenges which we have for the targeted drug delivery system. Now I'm coming to the new avenues, new field of the research, which is directed energy. What directed energy encompasses for the targeted drug delivery system involves nanotechnology, biomedical engineering, controlled stimulus to achieve the precise therapeutic effects, minimizing systemic toxicity. So they are the photons. They are the photons. They are the energy carriers. So the modalities, they go for the photodynamics. They have the electrical energy harvestings, nanoparticle carriers. So rather, depending solely on the chemical signal or the passive accumulation, it utilizes precise, externally controlled physical energy fields to manipulate, guide, and activate therapeutic agents. So you give them your drug, targeted drug, let them reach their spot, you give them directed energy. At that spot, they will transfuse into heat and they will release your drug. So that will reach on the precise location. When we talk about directed energy spectrum, now the directed energy spectrum is a lot of the spectrum. Whatever you see from the your radio frequencies, microwaves, millimeters, infrared, ultraviolet, X-ray, and gamma radiation. By the physics rule, we know this much still now. We do not know how much wavelength we still have in our nature because gradually physics is discovering the new. Still there, IR was not there because it was not visible. So an IR reason has come up. So now in future, what will come up is not known. So there is a spectrum. This all is spectrum in one or the other way can be utilized for the directed, targeted drug delivery systems. So you can use this all systems to deliver your energy and target them. And this technology has a good preciseness. So you transfer the energy, you target your molecule, you reach up to the site. When we talk about this high power energy systems, in case of defenses, we use for the target designation, neutralizing UAVs, aircraft. So for directed energy, we are using in the destructive way. So I'm talking on the reverse side of this because now earlier the systems was like you had to just fire it out. Now you have the control systems. You have now the better optics in physics. So you have the better optics. Now you have the tunable control systems with you. So what you can do, you can now tune in. So you can tune in your wavelength. So you can precisely deliver. If you don't know 2 watts, 4 watts, you do not do like 20 watts, do not know 20 kilowatts, you can deliver milliwatts. You must have seen that therapies, they are now using the biophotonic. They use your drug, they give you some of the lasers of the 650 or the red lasers. They are the examples that low level energies, they can be good. At higher level, they can be bad. So this high spectrum at present, widely developed systems are the high power microwave, high energy laser system, and another one is the particle beams, which still is emerging. No particular systems has been developed, but shortly the systems are going to have the particulate beam and the emerging beams so that you can have the pinpointed beams. So military, why we are concerned in military about this system? Because we use everywhere in the radar systems, in the radio communications, in our all systems. Now the warfare is quite changing from the laser systems towards the RF system. They are destroying each other. So from the military perspective, radio frequencies, deep internal tissue burns can be on the longer time exposures, ocular trauma mitigation, neurological protocols, brain traumatic injuries are there. So if you are continuously exposed to the violence for a longer duration on the defense part of systems, that's why the military is also concerned with this, and we are also trying to how to mitigate with this challenges. So what the systems involve, this all your directed energy systems involve is the external source. Now you have the sources, you have a machine, which you have the laser guided RF systems, you have the systems. Then the systems can go in your body and can create your acoustic energy, electromagnetic energy, or the magnetic energies that can pull your targeted molecules and can go to the can pull your molecules and make them to go to their diseased tissues. They can cause acoustic cavitation, they can cause vaporization, even surgeries, they are using the lasers for laser knife to cut the extra tumors, photothermal, photodynamic. So there are a number of mechanisms possible with this directed energy. So medically, it is a boom that we can use this system. Also, scientists have now discovered that blood-brain barriers can be crossed with the assistance of directed energy. So this all is a clinical perceptions that how laser ablations, laser can be used to ablate the tissues, extra tumor tissues, and how the RFs can be used in the different coherence, how can we judge the optical coherence of the eye and system. So lot of the systems, the pulse electromagnetic, how you can rotate this pulse so have your bone fractures can be recovered faster. So this all are energies. How you tune in the energies, they will help you in medicine in a larger way in a coming era. So there are number of interactions what they do with your tissues, is thermal effect, non-thermal effect, neurological effect. So n number of possibilities are with this wavelength. The beauty of this wavelength is that all wavelengths have their own different type of effects. So you can tune in what you want from them. So you have the lasers, you are superficial, you have the milliwave meter waves, high power microwaves, which have deep penetration. With this small spectrum, you can see how you can target, how deep you can target, what you want to target. So there is n number of possibilities you can use these systems. The same the direct energy can be used to dissipate the heat, photo dissociation, shock, generating a shock wave within the tissues, can be used for the diagnostic, for the photochemistry analysis. So you can have your imaging systems also based on the systems. You must have seen your confocal systems or your systems. They are all based on the 532 nanometer lasers. So they are already using this energy systems. But the systems, until are not more tunable, you need to be cautious because that can be deleterious as well. So you need to have the knowledge how to operate and what level you have to operate. So coming to the some of the systems, the laser assisted delivery system. Now the system, laser-based systems have been used to ablate you fractions laser. So already they are used in the medical sciences for the ablative fraction lasers where extra tumors have been removed using the laser knife. So they are being used for treating scars, actinitic keratosis, skin cancer, melanosomas, and vaccination. So most of the things already, few of the systems has been already in use. In biophotonics, they are already been used to deliver the topical, your topical carriers to penetrate deeper into the tissues. So they assist your drug to penetrate deeper, which is not possible for the slow process. So there are, again, with the directed energy, you have lots of possible. It has your penetrative depth, photothermal effect, photodynamics, phototherapy. So with laser, you have lots of play and flux to do with this system. You need to have the precise layer. You need to have the precise knowledge of that. The disadvantage with the laser therapies is because they are more being absorbed with the micromolecules in skin tissue, like melanin and your more micromolecules. That's why this system has still slight limitations, and they are being used for the topical applications because it is more absorbed on the skin, and going to a higher skin, it gets called burn. Then coming to the another part of the laser is a near infrared laser for the targeted delivery system. That also has the spectrum of 700 nanometers to 1,000 nanometers. So it also has the lot of the potential applications. It also has the most of the photo clearance, linkage release. It can go for the photodynamics therapies. It can have IR-induced hypothermia. So what they actually do, they take your carriers. They take your encapsulated carrier towards the one of the target organs. You let them reach your medicine towards the disease organ. Then precisely you fire on that your nano carriers or whatever carriers you have. The capsule will be taking the hypothermia reactions, and they will be damaged, and they will reach to your site. This NIR has the more advantage towards the laser is that laser has more absorptions as the laser macromolecules. But this NIR and the IR are slightly not been absorbed by your it has lesser absorption towards the melanin macromolecules. So that's why this is more useful than laser. Lasers are more useful towards the topical applications, but when we go for the NIR applications, this is more for the deeper penetration tissues. It also has the deep tissue consent. This can be used for the deep tissue combination therapies. So what you have, you fire your carrier, you deliver your drug, and give some energy so that can heal faster also. So it can be used as a dual therapy. So NIR is better than laser, but both have their own applications and way. This is some of the studies what I am saying. Use this one we have taken as a visible laser, another is a NIR laser. So this is the difference I am showing it to you. When you go for the visible laser, they are more absorbed on the skin, and there is a dermal inflammation. And if you see, there is a burn over there. If you see the lower panel, there is a burn. So these lasers are absorbed on the skin, and they cause burn. So, and when we go for the NIR, it penetrates deeper inside the tissue, bypassing your macromolecules and the most of the molecules. That's why this gives. And hence also that when we go for the visible lasers absorbed on the skin and creates a high temperature. Our NIR goes deep into penetration and has a lesser temperature profile. This is slightly part of our biodefense program, where what we are trying, we are giving this NIR laser to the brain just for the few seconds. We have just given them for how they are transferring energy. You can see how damaging it is, but how I can say, if I can control in a better way, I can use it for the therapeutic purpose also. So it has a dual angle. There is a limit and a set margin. You can go on this side or that side. So in rodents, what we did, we given them a NIR laser of the 2 mm spot for few seconds and a minimal wattage, which do not cause any thermal increase. We found the rats has a delayed emergence. Then when we given it for only just 20 seconds, there was reduced activity. When we given it for the 30 seconds, there was loss of movement and reduced activity along with the spinning and the locomotor circling. So you can see the technology has a high value that in a small fraction of seconds, they are transferring this much of the energy. So if we can fine tune them, how good can they be for the medical purposes? These are slightly the retinal effects of these lasers, which we have done. Will not go through this. So it also has the retina. For this retina, most of the lasers are quite damaging. So we need to have threshold for that. These are some of the 3D human tissues which we have obtained from the MedTech Corporation USA. We are using them for the laser for our own defense purposes, for the safety trial purposes, because we have our own Jawan posted at the different postings, and they have been exposed because now the lasers are being used widely in the warfare. RF are being used. They are been targeting different diplomats are been targeting using the RF microwave. So now the different games have been played. So we have to see our Jawans who are using these systems are also safe. So this is a part of our system. Coming to the RF. Radio frequency. When we call for a radio frequency, it encompasses 30 gigahertz to 300 gigahertz. What you have your mobile phones, they are also coming. So in megahertz, it penetrates 20 to 200 centimeters in the biological tissue. So RF responsive nano carriers are the smart containers, liposome missiles that contains energy transducers, gold particles. They can then again they go inside the tissue, create a hypothermia. This hypothermia melts their outer membrane. They release their drugs into systems. So now what they have come with the RF system is a small implantable device pre-filled with the multiple doses of medication. So RF, what they are using implantation, so that tiny reservoirs filled with the tiny membrane upon receiving wireless RF signals. So you have the some implants inside. Now you can control from the outside what to deliver inside. So now they are using this RF technology because RF can communicate your chip inside your body and outside. So this communication can set up. So it's a newer future where you can have this kind of drugs where RF will allow your chip to communicate and release the sustained release and select you and the controlled release of your drugs. So these are again the same pictographic of RF, how they can do it. Then again is the ultrasonic drug delivery system, so sonoporation. This electronic ultrasound, which you know, these are high. Now the system is coming is a high intensity fused ultrasound or the low intensity fused ultrasound. What they do when your molecule is traveling some portion with ultrasound, you can focus your tumor. Once your carrier reaches your tumor organ, you can give them energy, high energy, intense energy, so that they break down and mostly they reach your target sites. So they make a acoustic barrier to cross, and the it goes inside the tumor. So what achievement they have obtained is now the blood-brain barrier. They are able to cross this blood-brain barrier with this fused transducer. What they are using is a fused transducer where they are trying this drug molecules. They are giving them energy to make a cavitation. This cavitation make them in a bubbles, creating a bubble. That bubbles keep fluctuating and going expansion and thermal expansion and contraction. The thermal expansion and contraction makes your blood-brain barrier to open temporarily. Then the drug crosses, and with time, they have found that these systems can be restored back. So in 6 to 24 hours, this tight junction gets closed. So with using this ultra focus open blood-brain barriers, you can have these systems. Coming to the magnetic field delivery systems, you have here you have to have your super paramagnetic iron oxide or some metal oxide based drugs. You can have the metal based oxide drugs. It's like a pulling your magnet. You have some metals over there. You are pulling with magnetic energy. So at the target of tumor cells, targeting a tumor cells, you can target and pull them out. So they can be pulled at one place and can be released. So this is a magnetic targeting field. The another is a pulsed electromagnetic field targeted drug delivery. difference between them is it's pulling out. But with the pulse EMF systems, these pulse systems are more energy, having the more energy. So they keep on switching on and off and on and off. So you can have the longer, deeper therapies. So you can keep on and off. So you can have the controlled control unit with this line. So you can use this PMF systems in selective way and a controlled way to deliver. Also, what they do, if you have your rod carrier, so they have the pulse energy. So what will they will have the circular motion. So they can help your drug to reach to the spot. So if you so another purpose is you can reach your spot. So if you are unable to reach your spot, so you have the some red card, you ask them to circulate. In circulatory movement, they will drag also. So they will drag also, they will reach to the spot and then they will be released. I will finish up. So this is a complete, I'm concluding my talk. So this is still much of the wavelengths which I have spoken. Most of them are being using for the biological research purposes and they have been proven in biological processes. Some are being using medical technologies like ultrasound is a good option where ultrasound we are widely using. So it can serve both the purpose. What ultrasound does with you? You gives you the sonograph also. You know the drug also. So it is giving the diagnostic also. What now you call it therapeutics. So you have the therapy with the diagnostics. So you diagnose, you give the drug, the same time you destroy that. So this is by and large, all the systems have been proven. Their biological interaction with their tissues has been proven. And now just we have to wait how the systems are designed for the biological part of the system. And we can thank you, everyone. I rest here.
Sir has shared very insightful things today, but due to paucity of time and we have to start with the next session. And certainly all these ideas we'll be taking forward. And we already have a collaboration with Dr. Madhumita and of course that collaboration continues. So moving on, I think Asmita, please take it.
With this, we would conclude the proceedings for our inaugural session and move to the felicitation ceremony, where we express our heartfelt gratitude to all the esteemed dignitaries present. I request everyone to join me with a warm round of applause as we felicitate and honor their valuable presence.
I request Professor Ashish Mani and Professor Puniti Mathur, AIV, to kindly come forward and felicitate Professor Dr. Trimurti Velpandian. Thank you, everyone. Now I request Professor Madhumita Ghosh and Professor Mariam from AIP to kindly come forward and felicitate our invited speaker, Dr. Rajkumar Tulswani. Thank you, everyone. So as we approach the conclusion of this insightful and enriching session for day one of our workshop, I sincerely thank our honorable dignitaries, keynote speakers, and invited experts for sharing their knowledge, expertise, and visionary insight with us. Your contribution have greatly enriched this workshop and inspired all the participants, setting a strong, inspiring tone for the next 2 days. I also extend heartfelt gratitude to organizing committee, faculty members, research scholars, technical staff, volunteers, and all the participants for their enthusiastic involvement and dedicated efforts in making today's session successful and memorable. Thank you, everyone. So moving on, I invite all the dignitaries, speakers, faculty members, students, participants to join us for the networking lunch, for which you have been eagerly waiting. So it's my request for the participants to free yourself as soon as possible. Thank you. I request all the members to please sit on the front rows. Sir, we can start.
ये वाला?
Yes.
So good afternoon, everyone. With this, we'll move on to the next session of the oral and poster presentations. Here we are doing our oral presentation session. The judges for this session will be Professor Dr. Mariam Sarwat, Amity Institute of Biotechnology, AUUP, and Professor Dr. Nirupama Dhiman, Amity Institute of Pharmacy, AUUP. We now invite the participants for the presentation session to showcase their research work and valuable contribution. Our first participant is Ms. Nidhi Verma, IIT BHU. Also, please both the judges, please come on the dais. We would like to invite you to come forward and sit on the dais. So there will be a 5 minutes of presentation and 2 minutes of question answer session. 17 to 21. Good afternoon, everyone. I am Nidhi Verma from IIT BHU Varanasi, PhD research scholar working under supervision of Professor M.S. Muthu. So, as we know, breast cancer is a major global health burden and is most common diagnosed cancer among women worldwide. According to Globocan 2022 statistics, 2.3 million new cases with 670,000 deaths were reported globally. Despite advances in the therapeutic strategies for breast cancer treatment, these conventional therapies faces several challenges like non-specific distribution, drug resistance, systemic toxicity, and low biological compatibility. So to overcome these challenges, nanotechnology-based therapeutic systems is being evolving to for effective therapeutic applications in cancer treatment. Among these, exosomes are natural vesicles which have natural biocompatibility and intrinsic tumor targeting ability. So therefore, we have developed exosome-fused liposome-based nanoformulations titled its exosome-fused liposome formulation stability studies and thernostic evaluation for breast cancer application. My presentation is majorly divided into 5 sections. First, with preparation and characterization of fused vesicles, following in vitro studies, followed by mechanistic studies. The prepared vesicles were also evaluated for in vivo safety and tumor targeting efficiency and anti-tumor efficacy. First, exosomes were isolated from human blood plasma using a common technique of PEG-based precipitation method. Then the isolated exosomes were fused with prepared liposomes. Liposomes were prepared by using solvent injection method. Then the fused vesicles were formed using fusion method. 2 vesicles were formed, drug loaded, that is this one, PLV exolipo vesicles and diloaded vesicles. The prepared vesicles were having particle size in PDI in the nanometric range with high zeta potential and entrapment efficiency and drug loading efficiencies. FTIR was used to validate the fusion of the vesicles and retention of exosomal compartments. Further, morphological characterization was done using microscopic techniques. The microscopic techniques showed that the prepared vesicles were having spherical morphology with amorphous nature and crack-free surfaces. Storage stability studies were done at 3 temperatures, 25 degrees Celsius, -20 degrees Celsius, and 4 degrees Celsius. At all the 3 temperatures, the prepared fused vesicles were having more stability than the formation loaded with exosomes and liposomes. In vitro release study was done at 2 pH 5.5 and pH 7.4. At both the pH, the formulation showed the pH responsive drug release and also sustained drug release. In vitro studies were done in the MCF7 breast cancer cell lines. A cytotoxicity study showed that concentration dependent increase in the cytotoxic effect, in which the fused vesicles showed highest cytotoxicity among free compared to the free other formulations and free drug, that is palbociclib we used. Ross analysis also showed that increased oxidative stress was caused by the fused vesicles. Under apoptosis study, the increased apoptotic cell death was also showed by fused vesicles. Cellular uptake was quantified both qualitatively and quantitatively, and fused vesicles showed increased apoptotic increased cellular uptake as compared to other formulations. Further, we evaluated the mechanism of apoptotic cell death using these assays, caspase 37 assay and annexin VPI analysis. In this, both the studies, we can see that the cells are moving from life cell, that 4th quadrant. The cells are in 4th quadrant. Life cells are more, but in fused vesicles, this one.
more number of cells are in quadrant 2 and quadrant 3, that which is showing the fused vesicle effect, more apoptotic cell induction in the fused vesicle formulation. Further, in vivo okay. Further, in vivo safety evaluations were done in histopathological analysis and hematological profile and biochemical profile. All the fused vesicles formulation showed more biological compatibility as compared to the free drug. Tumor induction was done in SD rats using DMBA, and a tumor model was evaluated for tumor targeting efficiency, anti-tumor activity, and vascularity. Here we can see that the fused vesicles were having more tumor targeting efficiency as compared to the free dye and only liposomes and only diloaded exosomes. Here anti-tumor activity was evaluated and fused vesicles showed more decrease in the tumor size as compared to the other formulations. Vascularity study was also done to check the angiogenesis effect. And all the studies were done using photocaustic and ultrasound imaging. As in conclusion, exosome fused liposome vesicles showed enhanced stability, cellular uptake, biocompatibility, and anti-cancer activity with effective pH responsive drug release. Ultrasound and photocaustic imaging confirmed superior tumor targeting and significant reduction in tumor progression, highlighting their potential as a promising diagnostic platform for breast cancer diagnosis and therapy. These are references. Thank you. Yes, ma'am. Yes, ma'am.
Hello. What is the principle of this essay, Annexin VPI?
Ma'am, Annexin VPI is basically an assay which we use to it is a quantitative assay which we use to determine the late apoptotic cells, earlier apoptotic cells, live cell, and necrotic cells. So in this assay we get the results in 4 quadrants. Quadrant 1, 2, 3, 4, which is this one.
What is Annexin V?
Annexin V is a marker which binds to the phosphatidylserine serine of the biological membrane, and PI is a staining dye also, which binds to the membrane but in dead cell. So when the necrotic cells occur, so PI will bind to the dead cells and Annexin VPI bind to the live cells. So accordingly, we get the data quantitatively.
In the data according to the
Yes.
principle.
Yes, ma'am. There are 4 quadrants. Quadrant 1, 2, 3, 4. This one, this quadrant is quadrant 1, quadrant 2, quadrant 3, quadrant 4. Quadrant 1 is representing necrotic. Sorry. Quadrant 1 is representing necrotic cells. Quadrant 2 is representing late apoptotic cells. Quadrant 3 is representing early apoptotic cells, and this is representing live cells. So this one this is control. So the cells are in more live in quadrant 4, which is live cells. Then PLB only drug. The cells are in live cells, and then quadrant 3, that is early apoptotics. But PLB loaded exosomes and PLB loaded fused vesicles. So fused vesicle 1 is having the more cells in both quadrant 2 and quadrant 3, which is showing that our developed vesicles are having more efficacy as compared to control, pre-drug, and PLB loaded exosomes.
So what is the conclusion of your study?
Conclusion
according to this Annexin VPI assay.
Conclusion of this study is that when we are using the exosomes with liposomes, we have prepared liposomes using the lipids TPPC, DSPE, PG, and cholesterol. So when we are preparing pegylated liposomes, and exosomes also have natural intrinsic intrinsic tumor targeting ability. So by combining both these ability to target the cell, it is going more, it is getting more to the tumor cells and getting more accumulated there. So by these effects, it is showing the more efficacy of the developed formulation.
Yes. Okay, just let me know. You have said that exosome
Yes, ma'am.
plus vesicle liposome,
Yes,
right?
ma'am.
So, what do you mean by vesicle liposomes?
Vesicle,
Liposomes.
vesicle liposomes, in what sense, ma'am?
You have
Yes, ma'am.
right,
Yes, ma'am.
and the liposomes.
Yes, ma'am.
You have used the term vesicle.
Yes, ma'am.
What does that represent?
Ma'am, because we have used the term vesicles because exosomes are extracellular vesicles which are released from the biological fluids, and liposomes are also a type of vesicles, bilayer vesicles. So that's why when we are using that, so to write in our research that's why we are termed it as hybrid nanovesicles.
Okay, so you also have shown that when you have just made this exosome vesicles with
Yes.
liposome, right?
Yes, ma'am.
So they are showing good effect,
Yes, ma'am.
right? So as such, if I will ask you, what is the action or mechanism of exosomes that they are, helping as anti-cancer?
Ma'am, as in general, I will tell that exosomes have some natural targeting ability because they can target to the cancer cells by receptor mediated endocytosis. That study we have not done yet, but there are some receptor mediated endocytosis like creatinine mediated endocytosis, clavulin. So these are some mechanisms that through these mechanisms exosomes target to the cells specifically.
Thank you.
Thank you.
With this, I would like to call Vaishnavi.
Good afternoon, everyone. I am Vaishnavi, a final year master's student from Amity Institute of Biotechnology, and here I'm going to present my work, Green Synthesis, Characterization and Therapeutic Applications of Nanoparticles Derived from Dalbergia sisal Leaves, under the guidance of Dr. Pramila Gupta. She is the Professor and Dean, University School of Biotechnology, Guru Gobind Singh Indraprastha University. We all know nanotechnology has been gaining a lot of recognition these days, and nanoparticles are materials with size dimension less than between 1 to 100 nanometer. At this scale, they exhibit some unique physico-chemical properties, which are not usually seen in their bulk form. The synthesis using green methods is paving a new way for the scientists to look for more eco-friendly, safe, and sustainable approaches by using less chemicals as possible. A type of nanoparticles that is gaining recognition is the carbon-based nanoparticles, which include carbon dots, graphene dots, carbon nanodots, because they show high fluorescence, biocompatibility, and drug delivery potential. There are various characterization techniques which are used to validate their potential. And here I have used Dalbergia sisal, which is also called as Indian rosewood or more commonly shisham. It is a medicinal plant which is widely known for its antioxidant properties, and its leaves have been proven to be beneficial for synthesis of nanoparticles and potential biomedical applications. So this study included 3 main objectives. First was to establish a low-cost, eco-friendly method for nanoparticle synthesis using these leaves, to provide a detailed physicochemical characterization of these nanoparticles, and to demonstrate their biomedical relevance in particularly dermatological sector. This was the methodology which was used. I collected the Dalbergia sisal leaf sample in the university, that is DGS IPU. Then I washed it using 1% labeling solution, and I left it to air dry for 15 days. After 15 days, I ground. This was the experimentation. The third trial was the optimization trial. And this is a pictorial representation of that. These were the results of the characterization. In this result, the DLS, we got the perfect size of 10 nanometer, which is why I call them as carbon nanodots. Through XRD, we were able to understand that there is a broad diffraction peak, because of which they are amorphous in nature, and other characterization techniques further confirm the successful synthesis of green carbon nanodots using these leaves. Moving on, this was photoluminescence analysis, which proved that they are fluorescent enough to be used as an application in their optical sensing and biosensing. I then formulated them in gels to check whether they are compatible or not. So they provide some fluorescence when they are mixed in the gel also. This is their spreadability. And then I tested them for their UV blocking and SPF potential, where I tried 4 different combinations. The CNDs alone, however, gave a good SPF value, but when they are incorporated in this carbopol gel, they somehow don't provide a good SPF value. So I tried combining it with a standard commercial product with a known SPF. So with a 3 mg concentration in the standard product, we got a rise of a slight rise in the SPF value. Then I confirmed the antioxidant potential of these CNDs using 3 assays, DPPH, ABTS, and FRAP. Through DPPS, I was able to understand that though these have an antioxidant power, but not fully against DPPH, as compared to ABTS, they have a higher antioxidant power against ABTS. Lastly, I performed FRAP, where I was able to understand their ferric reducing capability, and where the FRAP value was expressed in forms of Trolox equivalence. So through this, I was able to understand that we can form carbon nanodots using these techniques, and these can be used for various applications like antioxidant, anti-inflammatory, antimicrobial, UV protective, etc. These are some references. Thank you.
Hello. Yes. So, what do you mean by nanoparticles first?
Particles that have size dimension between 1 nanometer to 100 nanometers.
Okay. Now, come to the characterization, na? Zeta potential. Yeah. What is the importance of zeta potential in nanoparticles?
It is important to understand the charge which is present in the nanoparticle to determine their application in further sectors. So here I got to know that it is negatively charged, which could be useful in dermatological applications. Like it has to be tested further. So
Wait, why it is useful in dermatological only?
I've just tried one sector for now for like them because it has antioxidant potential. So I thought of testing it into like the dermatological industry first.
Okay, you talked about antioxidant potential, right?
Hmm.
Come to that slide. You have said that this particular, it is showing effect there in DPPH, right? In different three you have done.
Yes,
Na?
yes.
So how you can say that it is showing the antioxidant potential there in DPPH?
By the IC50 value. IC50 value is the value at which at the concentration at which 50% of the radicals are neutralized or scavenged. So through this, using the standard assay, although the IC50 value shows a good potential, but with the carbon nanodots which I have synthesized, it shows a moderate effect because they do not have these CNDs do not have hydrogen donating capacity that much, which is why they show a better antioxidant power with ABTF.
Okay, come to only this, yeah, DPPI. So how you can say that this is, okay, this is antioxidant property you have seen, right? What standard you have taken to compare for the comparison?
Ascorbic acid.
So by comparing with ascorbic acid, now just let me know whether your practitioner or nanoparticle is better than that or not.
Against DPPH, it's not that good. But against the others, it shows better antioxidant activity. Thank you, ma'am.
Thank you, Vaishnavi. With this, I would like to call upon our next participant, Anshika. Please come forward.
Good afternoon, everyone. My name is Anshika Dera, and today I will be presenting my work on phytomediated synthesis of nanoparticles from Delbourgia bark for antimicrobial applications under the guidance of Dr. Pramila Gupta, Professor and Dean USBT GGS IPU, and Dr. Abha Kumari, AIB Amity University, Noida. So Basically, nanotechnology is a rapidly emerging field. Okay, ma'am. So essentially, I focused on utilizing Delberger bark for the synthesis of carbon-based nanoparticles using two carbonization-based approaches. First was from a hot air oven, and the second was from muffle furnace. So in hot air oven-based carbonization, I focused on synthesis of carbon-based nanoparticles by thermally treating under lower temperatures for controlled durations. And this is the result I got for for the nanoparticles under 3 hours in hot air oven based carbonization. As you can see here, we got result from characteristic absorption peak in UV analysis and got the size of 52.53 nanometer during the DLS analysis, along with a good zeta potential indicating good colloidal stability. Along with that, we did other characterization analysis, which indicated the successful synthesis of carbon-based nanoparticles using hot air oven-based carbonization. Along with that, the other method I used was muffin furnace-based carbonization, which essentially involved thermally treating our material, which was Delberger bark, under higher temperature for rapid carbonization. After that, we also, did the characterization of it, which indicated the successful synthesis of carbon-based nanoparticles from Delberger bark using muffle furnace-based carbonization. Essentially, these two, after that, we performed the antimicrobial assay of the two nanoparticles made from these two methods. So this was done using two methods, this diffusion method and a minimum inhibitory concentration MIC assay. So using the disk diffusion method, it was done against two strains, gram-negative E. coli and gram-positive Bacillus subtilis. So as you can see here, in hot air oven-based carbonization, the carbon-based nanoparticle B3 shows demonstrable zones of inhibition, albeit it can vary from concentration to concentration. And in muffle furnace-based carbonization, the carbon-based nanoparticles produced from this method, on the other hand, does not show that demonstrable of zones of inhibition, ultimately showing minimal or negligent antibacterial activity. This eventually suggests that a higher temperature led to the degradation of surface functionalization groups in our Delberger bark that could be essentially responsible for our antimicrobial potential. I also performed the minimum inhibitory concentration assay along with that, in which the B3, which is our carbon-based nanoparticles produced from the hot air oven-based carbonization method, showed the good MIC value of 500 microgram per milliliter for both E. coli and Bacillus subtilis. Meanwhile, the carbon-based nanoparticle produced from muffle furnace-based carbonization method, BMF2, showed a minimal or negligent antimicrobial activity. As we can see here, ampicillin here was used as our standard comparison. In this, E. coli showed the MIC value of less than 0.9 microgram per ml. And in Bacillus subtilis, ampicillin showed the MIC value of 125 microgram per ml. So in conclusion, the study demonstrated the synthesis of carbon-based nanoparticles from Delberger bark using two carbonization-based approaches and eventually evaluated the physicochemical properties and their antimicrobial efficacy. So we employed two carbonization-based approaches. First was our hot air oven-based carbonization, which was done on lower temperatures for controlled duration. And the other was muffle furnace-based carbonization, which was done on higher temperatures for rapid carbonization. And as we can see here, hot air oven-based carbonization was able to be our most favorable formulation due to its high synthesis yield of 83.8 to 85.18%. Its well-defined UV absorption profile, photoluminescent properties, and reproducible batch-to-batch performance, along with a good antimicrobial activity. While we saw muffle furnace-based carbonization also produced good nanoparticles with small particle dimensions and similar physicochemical properties, but was ultimately not able to be successfully applied in antimicrobial applications. So thank you for your attention. And these are some of the references I employed. Thank you.
Hello, am I audible?
Yes.
So come to the results of antimicrobial assays. Yes, this one. So explain which one is better. Which one is better? The other slide, this diffusion. Explain along with the figures. Yes. Explain with that of figures, which one is better.
We focus on the diffusion method. We can see here that our carbon nanoparticle produced from muffle furnace-based carbonization method did not show any zone of inhibition. The only zone of inhibitions you can see here were produced from ampicillin, which is our positive control. On the other hand, when we did the diffusion method for carbon nanoparticle produced from hot air oven-based carbonization, ampicillin.
Yes, we are not able to see it clearly. Which one is ampicillin?
Here, ma'am. The AMP showing.
Okay. So why this ampicillin is different in on the other different figures? Why not it is on the same position?
That was slight confusion due to a marking, but you can see here AMP is written.
Okay, now explain it.
So as we can see here, we can see a good zone of, not very good zone of inhibition compared to a positive control, but there were demonstrable zone of inhibition in this diffusion method done against E. coli and Bacillus subtilis compared to ampicillin. On the other hand, when we did the diffusion method for carbon nanoparticle produced from muffle furnace against E. coli and Bacillus subtilis, we could not see the same results. Albeit no zone of inhibitions were formed except for ampicillin, which is our positive control.
Okay, so you want to say that this is not effective. This you ask.
Muffle furnace-based carbonization, yes, ma'am. I mean, there can be certain other applications that can be explored using this approach, but we can see here for antimicrobial based applications, lower temperature based carbonization is more preferred because it was able to retain the functional groups that are required for its antimicrobial potential.
So what other experiments you have planned to further see this?
I will have to study more about it.
I have only one question, right? You have said that in muffle furnace, there is no activity, no antimicrobial activity.
It did show good physicochemical properties on its when we did its characterization, but when we did the antimicrobial application assay, it did not show any significant activity.
Why?
That is, ma'am, because muffle furnace-based carbonization was done under very high temperature of 300 degrees Celsius. So I guess it led to the degradation of the functional groups that are required for the antimicrobial potential.
So functional group of what?
Function. I did the FTIR, I did show degradation of lignocellulosic groups that I think might be responsible for it.
Come to the title. Yeah. What is the role of the bark?
Bark is, ma'am, also
Source of what?
Of the carbon nanoparticle synthesis. Thank you, Anshika. Now I would like to call upon our next participant, Marusha.
Good afternoon, everyone. My name is Maru Shamir, and I'm currently pursuing my master's from Amity Center of Nanomedicine. And today I'm going to present on the topic development of quercetin-based bovine serum albumin nanoformulation and evaluating its efficacy against breast cancer using in silico and in vitro methods. So in this presentation, I will be talking about the objectives, introduction, the in silico studies, preparation, and this work is currently under process. So the future work plans with this work. So my objectives are to evaluate the in silico efficacy of quercetin against breast cancer target proteins, to develop and optimize the quercetin-based nanoformulation, and to evaluate the in vitro efficacy of developed nanoformulation on breast cancer cell line. And my cell line is MCF7. So as you all know, cancer is a disease that is okay. So the target protein that I chose was SMYD2, that is overexpressed protein, and it was the PDB data of the protein. So this protein, the role in normal cells of this protein is that it methylates histone at lysine 4, and it actually helps in fine tuning of the gene functions, and also fine tuning of P53 as well as retinoblastoma. And in cancer cells, what happens, it suppresses these tumor suppressors like P53 and all, and due to which causes the cancer for uncontrollable proliferation. So how it actually works, it uses the methyl group from the molecule, that is SAM, and it is attaches it to lysine residue on the target protein. This is actually the schematic how it works, how it transfers the methyl group to the target protein. So the in silico studies were done. Actually, I have done the docking of various drugs, and out of these, I selected quercetin because my binding affinity was very much good. I also got the good binding affinity with hesperidine, but in my studies, I still chose quercetin because the result of hesperidine in the studies which I researched and studies was not that much good in vitro. That's why I moved forward with quercetin. So quercetin is a flavonoid which is present in apples, berries, and onions, and it helps in the inhibition of the proliferation of cancer cells. It helps in apoptosis, and molecular studies also suggest that quercetin can interact with the active site of SMYD2 protein. It can inhibit the methyl transferase activity. However, the poor solubility and bioavailability of quercetin, due to which I chose to form the nanoformulation-based drug delivery system, which was BSA. So BSA acted as a carrier here. So these were my docking results, and my docking results showed it was the blind docking done here. And my binding affinity was -10 here. And then site-specific docking was -10.36. And these were my post-docking analysis 2D interactions were done here, which showed many interactions were with conventional hydrogen bonds, so that we can see the bonds were more stable at the active sites here. And after which I moved forward to the nano formulation, which was BSA nanoparticles. So BSA nanoparticles are natural, biodegradable, and biocompatible. That's why I took them as a carrier. So preparation was done by desolvation method, and the work was continued for optimization of BSA nanoparticles. And future work plan, actually, my characterizations have been done, and I got the good results in DLS. It was almost 60 nanometers. I have not shown that here because my work is under process still. I will be completing in 2-3 weeks. So that data is not shown here. After that, I will do the in vitro studies. So till now, I have done this much work. Thank you.
Okay, come to the in silico study. How many, you said that these phytoconstituent you have selected. On what basis you have selected these many phytoconstituent?
These many drugs. Actually, we were given first we were given the time to choose the protein that we want to target. So, and these were the available drugs. They let me to choose actually. So I go through the studies and study the which drug can be used as a targeted on the selected protein. So it got aligned with the selected protein, SMYD2 protein.
That is what I am asking. That why you have selected these many only? Only you have done with only these 8 molecules.
Yes, ma'am. 8 drugs. I have docked these 8 drugs on SMYD2, and out of them, I have chosen quercetin.
Okay, come next. Thank you. Next.
Acha, ma'am. Sorry.
Okay, आगे जहाँ पे हाइड्रोजन बॉन्ड लिखा है
Acha.
so here how you can say that your this is the structure of
Sir, yeah, ma'am, it is the interaction of this my ligand and my protein, my drug with the protein.
this is the structure of quercetin.
It is the structure of my protein. These are the docking results.
Come to that slide only. This is the structure of quercetin. Yes or no?
I just got confused. This is my this is my protein here.
Yeah.
Yes, protein along with. It is the 2D, it is this, the docking done here, and it is its 2D interaction. It is, this one is 3D one, and
Yes.
it is showing the, okay, it is showing the interaction of the drug with the protein at the active site.
Okay, how you selected the active site?
Active sites, I selected it from the already present papers and research.
Okay, the protein which you have selected, it is having any ligand with it?
Ligand instance.
Actually, you have to clarify, right? Okay.
Yes, ma'am. First, I have done blind docking, and after that, I have done site specific as well.
Next time, present whatever you have done.
I have done that.
Thank you, Marusha. With this, I would like to call upon the last presenter, Ishita.
You see?
Good afternoon, everyone. My name is Ishita Bharti, and I am pursuing my PhD under Professor Dr. Seema Bhatnagar. The title of my presentation is Design to Delivery, Plant Anti-Angiogenic Leads for Targeted Nanomedicine. So starting with what is angiogenesis? Angiogenesis is formation of new blood vessels from existing vascular structures, which is essential for tumor growth. Okay. Okay, so my work is done in several parts. First, we have identified the plant Clerodendron Unfortunatum. The reason for identifying this plant is because it has reported literature talking about anti-inflammatory and anti-cancer properties, which help us induce that it could also be a potential for having anti-angiogenic activities in it. First, we macerated the plant and we made the extracts from it. The extracts are made by 4 fractions. The first fraction was the methanolic fraction, which is highly polar. Then the next one was hexane, which was non-polar. Then it was ethyl acetate, and then last was chloroform. All the extractions were individually tested in the CAM assay. The CAM assay study can be said as a gold standard for anti-angiogenic results. Out of all the 4 extractions, we got the best results in the ethyl acetate extraction. As we can see here, there is a good vascular structure in the negative control, and as the dosage increases, we see there is a depletion of the vessels. And compared to positive control, which we used as quercetin, which is proven an antigenic agent, we have seen that increased dosage is giving us increased anti-angiogenic effects. Further, we took the extract for phytochemical profiling, where we got our LCMS analysis done. LCMS analysis showed us multiple peaks, which were polar and semi-polar. And we got a key peak at 23.43 point, which we have interest in furthering our studies to. The next, for volatile compounds, we went forward with the GCMS analysis. 2 major compounds were identified in this, which were palmitic acid and phytol. Palmitic acid and phytol have both been reported to have antioxidant and anti-inflammatory activities, which are a crucial process of the angiogenesis modulating function. From the results we got in the LCMS, we used 2 compounds, plus minus pinoresinol and acacetin. These were then docked with VEGF R1 and VEGF R2. VEGF is important receptor pathway for vascularization promotion, and it is very important in the angiogenesis process. So that's why we chose this receptor as our point. Both of these compounds, we first docked it with benzamide in 3-HNG and sunitinib in 4-ADD, which are reported drugs for VEGF to stop the VEGF promotion. And compared to these drugs, we got good results in plus minus pinoresinol and acacetin as well. We would like to infer from this that considering the pinoresinol is a lignin and lignins promote the endothelial vascular growth, so it might be a good fit for furthering our anti-angiogenic study. Through all of this, we have concluded up until now that by doing polarity-based root extraction of Clerodendron unfortunatum, we are seeing a good antigenic activity in the ethyl acetate extract. The LCMS and GCMS also gave us good bioactive metabolites list, out of which we could identify pinoresinol and acacetin as our lead molecules to further our study. Even the molecular docking confirms that these binding affinities of the phytochemicals with VEGF-R1 and R2 are a good lead for furthering our angiogenesis inhibition. So with this thing in mind, we have an ideation that we can further our study once we have done our assays for specific compounds with purified compounds, we can further our study for nanomedicine translation. For this, we have four targets that we can do. First would be the phytosomal nanoencapsulation target, considering that quercetin and pinoresinol are both hydrophobic, thus it is well limiting oral bioavailability. So we can further it by nanoencapsulation. The second would be the VEGF targeted nano delivery. So for this, we can use the target as cyclic RGD, which targets alpha V and beta 3, which are generally overexpressed 5 to 10 folds in the tumor vasculature as compared to the normal vessel structures. The third we could go with is the combination nanotherapy, where we could co-load our extract or our compounds with VEGF siRNA into lipid nanoparticles, so that we can have a synergistic effect, which can be anti-angiogenic as well as angiopreventive. The fourth pathway that we have chosen could be the green synthesis of metallic nanoparticles. So there has been recent studies on clerodendron and phosphonatin, which it is being used to biosynthesize silver nanoparticles, which in itself can show VEGF inhibiting activities. So we can do a combined activity of this as well. So this could be the proposed work plan for the future. These are my references. And I would like to acknowledge ACNM for giving me this opportunity to present. Thank you very much.
Okay, Ishita, come to the in silico activity first. Back. Okay. First of all, I'm not asking from here, but my simple question is that you have done the LCMS of which particular extract?
Ethyl acetate.
But you have not given it in the slide. At the conclusion, you have given it.
I'll keep that in mind.
Right? Okay. So in LCMS, which library you have used?
Ma'am, the library was provided by the people we got it done from.
So you have not asked them.
They give us a list library, so it was given by them only.
So how many compounds they have listed there in the library? You must have got the result now.
Yes, ma'am. Ma'am, there were about 15 to 20 about there were compounds listed in the library.
Okay.
So out of that, we choose to go forward with these two.
Why?
Ma'am, as I said, for pinoresinol, since it is a lignin and is endothelial growth promoter, we have done docking for almost all of the compounds that were present, and we chose the compounds that showed a good docking score of at least -8 or better than that. And from then, we chose these. We also got good results. We had a list of compounds where we had good results, which were like scutlerin, diacetyl, bicillin. We had a complete list of compounds, out of which we think it's better to go forward with the quercetin and pinoresin.
Okay, which reference you have taken to for this? This is better. Yeah.
Ma'am, so there is already study done on pinoresinol, which is showing that decrease or the induction of this ligand is showing decrease in the endothelial growth. Same goes for a quercetin. A quercetin has also been reported for anti-cancer activities on its own. And since our goal was to go forward with the compounds or the plant which shows anti-inflammatory and anti-cancer activity to prove anti-cancer.
The literature you have just taken that.
Yes, now out of all the compounds that we identified, and that got good docking results, from there we went to the literature and in fact which good compounds we could take. Thank you.
So what are the other angiogenesis assay you have done other than CAM?
So ma'am, first we did CAM, then we did go forward with an MTT assay. Ma'am,
results of MTT assay.
Ma'am, I don't it was before I prepared the presentation, so I could not put it in the presentation on time. But we did get a good result for MTT assay for ethyl acetate as well.
before LCMS, must have done that MTTSA also, no? Because not only CAM is not the only decisive factor.
Yes, ma'am.
Yeah. So before going for LCMS, you must have done
Yes,
that
ma'am. I
MTTSA.
just could not put it for this my mistake. I should have put MTTS as well. We also went forward with the scratch essay.
Okay. So MTTSA is done on which cell line?
Non pancreatic cell line, PANC1 and MIA PACA1.
Okay. But we can't say that these results are conclusive unless we have seen those results as well.
Yes, ma'am. I'll make sure to put them next time. Thank you. Thank you, Ishita. With this, we come to the end of the presentation, oral presentation session. Thank you all the participants for their active participation in this session. We would also like to thank Professor Mariam and Professor Nirupama for accepting our invitation to be the judges. I appreciate your consideration for this invitation. With this, I now invite Professor Seema Bhatnagar to felicitate Professor Dr. and Professor Dr. Chansi to felicitate Professor Mariam. To felicitate Professor Mariam Sarwat. Sorry, Professor Dr. Seema Bhatnagar, Dr. Chansi, and Professor Dr. Madhumita Ghosh to felicitate Professor Mariam. They now request Professor Dr. Madhumita Ghosh, Professor Seema Bhatnagar, and Dr. Chansi to felicitate Professor Dr. Nirupama Diwan and Dr. Deepika Chauhan as well. Thank you, everyone. With this, I would like to also make an announcement. The evaluation for the poster presentation will be done tomorrow. And with this, we'll also proceed to the workshop session. हाँ जी, हाँ जी सर। हाँ जी। May I now request IT host to please close the session or Zoom link as well. Sorry, just a second. Before this, we would like to, I would like to hand over to Nilanjan for presentation.
वीडियो ये करना पड़ेगा वीडियो
ये करंट ही है इस यहाँ पे और
हाँ यही है। टाइम चला गया।